Good morning, all. I think I'm lucky to start the session. My communication will be very well followed because now my mind is very fresh. Please help to display the slide. Yes. Before we display the slide, it is about a case we faced last year, intoxication of cows after grazing sorghum crops. I think some of you in Rwanda heard about this in the paper news, right? It is still opening. So you invited me before we get prepared. Eh? Maybe I can start by introducing myself for those who don't know each other. I am Justin Kabera. I work with the Rwanda Forensic Laboratory. I am in charge of chemistry division. In the chemistry division, we deal with drug analysis. We have two units. One is in toxicology, where we deal with samples from autopsies, sometimes, or samples from drug takers. Another unit is with chemistry and drugs, where we can analyze physical drugs like narcotics and other chemicals. So yesterday I was here representing our DG. Today I am the speaker, and tomorrow I think I will be teaching the same topic of toxicology in different sessions. So I think some students will be together at Rwanda Forensic Lab for a session on toxicology. Thank you very much. So the topic is sorghum poisoning in Kato in Kere district. Eastern province of Rwanda, a case study. Go to the next slide. I would like to to show you where Kirehe is. Next slide. I can use this. Oh, sorry. So, in red color, this is the Kirehe district. Is a district border with Tanzania. This is the Kirehe made bigger. You can see in the yellow the road from Kigali. This is the Rosoma of the border with Tanzania. So, what happened the last year? It was in November 2021. A farmer from Kirehe reported a sudden death of his 13 cows after grazing in nearby plot having immature sorghum. It was, for us, a first experience because we didn't see before a case like this, where cows can graze young crops of sorghum and suddenly they die. It was a new case. According to the statement, the cows was healthy before, and the owner was shocked to lose such number, and he reported it to Rwanda Agriculture Board. So we have been contacted by Rwanda Agriculture Board to help in analysis. Those are the pictures of dead cows after grazing these young crops of sorghum. The veterinarian from RAB, Rwanda Agriculture Board, collected the samples, urine, blood from heart, ruminal content fluid, and the leaves of immature sorghum to submit to Rwanda Forensic Lab for analysis. Because for us it was the first case. Before we didn't know that after grazing such crops, we can find intoxication. We started by searching in the literature. And in the literature, we found that the intoxication is possible. And many cases have happened in Brazil, in some many areas of where farmers grow cereal like this. So in such situation, where cows are grazing crops like this, there is a possible intoxication. So the farmers they must pay attention and not allow access of the cows to young crops. So according to the literature, we found that sorghum are recognized to cause nitrate and hydrocyanic acid. Hydrocyanic acid is also called prussic acid. Poisoning for ruminant. Ruminant is, in our language, is Vincent. Ruminant. In Kuza. Yes. So any stress of a plant which causes abrupt stoppage of growth can also contribute to nitrate building up. Drought, herbicide damage, even late afternoon wilting. So the area of Kirehe is known to be dry. There is a severe drought all over the area. Few rain. So that is possible that the crops grown in Kirehe, it is possible to build up the poisons of nitrates. So, still, according to the literature, nitrate poisoning is caused by a consumption of excessive amount of nitrate or nitrate from grazing crops. Nitrates are higher in young plants. That is the information collected from literature. And if you have seen the previous picture, those crops are very, very young. That means when it is a mature crops, the content of nitrate is less. Prussic acid also is higher in young plant than older, but its concentration more in leaves than in stems. Mechanism of toxicity. When talking about the toxicity, how the toxicity occur, after ingestion of nitrate, the nitrate ion are reduced to nitrate. Nitrate is more toxic than nitrate. In the rumen is a part of a stomach of ruminants, forming metaglobin, which results in hypoxia. Metaglobin, what is it? Is a dysfunctional form of hemoglobin that is incapable of transporting oxygen in body tissue. Hypoxia means low oxygen supply in bodily tissue. If that happens after ingesting nitrate, then nitrate is converted into nitrate and come up with metaglobin and hypoxia. Then, this is what I explained in the previous slide. So, nitrate toxicity, chronic and acute situation. It may be what happened, it was acute intoxication, but it may be also a chronic intoxication, where we have found abortion or weight loss, reduced milk production, not enough for milk, and other animal performance issue. So we may have a cow with those signs without knowing that it is caused by nitrate poisoning. So in acute cases, nitrate is absorbed into the red blood cells and they combine with hemoglobin, oxygen-carrying molecule to form metaglobin, which has been explained earlier. Metaglobin cannot transport oxygen in the body, ultimately causing the animal to suffocate and die. What happened in our cases? So, plant factors favoring nitrate of poisoning. So, it can be a plant species. Some plants are capable to build up nitrate. Some plants. There is also the part of plant. In our cases, the young plant, a part close to ground, has more nitrate than the part which is upper. A stage of plant. A young plant is more, have more nitrate than old plant. Weather conditions, even water, because if it is the area where you use fertilizer, water can bring nitrate in different area of environment. So, those are the information from the literature, and even the method got from different papers. So we used, for nitrate analysis, we used the method of Nelson-Casbray, and for cyanide, which is prussic acid, we used the method of Ferg, Anger, and Pickrate method. And for quantitative analysis, and the Lamer method for quantitative determination. Those methods are described in details in the protocol. Result interpretation. This table is to help to interpret the results. So from 0 to 0.4 up to 0.44, generally the crops are safe. But when you see up to, this is in ppm, this is in percentage. So when it is upper, other than 1.77, it is danger, not to feed. But we found that in those immature crops, we found that the content of nitrate was 3.5%, the double of 1.77, which explains the sudden death of cows. You can see that it was high concentration compared of these guidelines. Here we say that 1.7% not fit. And we found that in our result, we found the content for nitrate at a percentage of 3.5. So this explains the sudden death of cows. The method of cyanide, we didn't find cyanide in the plant. No cyanide found. Treatment. Last year, someone in the previous summer school asked me how to be treatment, because I used to talk about intoxication, and no treatment. So for now, I managed to find the treatment. So when this happens, you have to treat by intravenous injection of methylene blue. I checked in the FDA, that product is not accepted, no longer accepted, methylene blue. That means there is, that means there is an alternative product. So better to consult veterinarian to help in such conditions. So concluding by how to prevent nitrate poisoning. Do not allow hungry cattle access to high nitrate forage or pastures. Do not allow access, if you have your cows, do not allow access to young crops of any plant. Observe cattle frequently when you turn them into a suspect feed or pasture to detect any sign of toxicity. The cows may escape and go in the pasture, and then we not know where they have been. So if you don't know where they have been, check the sign of toxicity. So always give a stock access to fresh nitrate-free water at all times. Cows like to drink water, so you have to give them fresh water, nitrate-free. Those are some of the measures to be taken, but the water is always recommended when it is to detoxify cows after grazing. This is the end of the presentation. Your question and comment are welcome. Thank you very much.
Yes, thank you, Justin. You heard it. Anyone who's interested to learn more about this topic, cow sorghum poisoning? Any comments? Oh, there's one.
Yeah. Thank you very much for a nice presentation and very interesting topic. Yeah, I have some few comments and even question on these cases that happened on sorghum. We've seen that especially on methodologies used to take for sampling. I'm not sure how the sampling has been done. whether it is on a sorghum that has been after ruminant or the sorghum on field, how the sample has been taken. We've seen that it is highly concentrated on immature sorghum. This is the cases even that can happen on cassava, especially where consumed or with the ruminant. The same cases raised, as we know that cassava also contains cyanide. Yeah, it is similar cases that can happen. I also confused on the results that happened. The result happened, it was 3.5%. Yeah, thank you. Is this, that standard, is it from literature? Yeah, I have been confused whether the standard based on, this is the new cases in Rwanda as you told from the starting of this presentation. Where the standard has been from, is it from literature, is it from where? We have seen that for immature, maybe it's 1.5 remits. I don't know whether it is not allowed to be consumed.
I understand the question.
Yeah.
Thank you very much. Starting by the example of cassava you mentioned, cassava also has, it is the one, is the first to have a high concentration of hydrogen cyanide. But before consuming cassava, when it is human, they start by process. There is a process to remove cyanide. So about this case, there's a recommendation, I found them from literature. These are the guidelines. We had the result and the north. So we try to, this is from a paper. These guidelines from a paper. After having this result, we managed to find the reference where you can compare. And fortunately, we go to these guidelines, and where I found that above 1.77 is not to fit, and when we found that we have 3.5, it was automatically immediate that the cows must die. But this was from the literature, is a paper. If you want, I can give you the reference. Thank you very much. Another question, he still have a question? Maybe I didn't understand well or his concern.
Yeah, thank you. You replied, it's just I want to just continue with the questions. In literature, I didn't see the lethal dose concentration to mean in terms of consumption of those immature sorghum or any other related plants. Lethal dose, I am asking, lethal dose, to mean at least.
My reference here, it depends on the crop, you see? In my case, is sorghum poisoning, and the literature is informing me that this is already a lethal dose, you see? That means if you check for every crop, we can find a lethal dose. Maybe even for cassava, we have a lethal dose, which I don't have in my mind, but you can check and found. So I think for every crop there is a little dose. This one is specifically for sorghum.
There is no, thank you very much. Another comment, it takes a long time, but I'm sorry. Specific, is this toxicants, it contains, there is any specific organs where it poisons, or it can poison various organs? As we know that some toxicants that are specific, others that are systematic, they can act on, they can be on different organs, poisoning different organs. Does this really has a specific organs where it poison?
Human toxicity in the organ. So let's come on toxicity, this part. It is said that the nitrate will go to the blood cells. Your question will be answered by this mechanism of toxicity.
Yeah.
And then, nitrate ion are reduced to nitrate ion in the rumen. Rumen is the part of stomach of ruminant. That is an organ of this cow. And from there is the formation of methemoglobin. Methemoglobin, which is the dysfunctional form of hemoglobin that is incapable of transferring oxygen. So we can say rumen, blood, probably with also the liver, because the blood pass by the liver also for metabolism.
Yeah, thank you very much. But maybe there can be by transformation, in case of chronic cases especially,
So
I think there can be by transformation, and sometimes the sign can be lost in terms of forensic, where but once by transformation is happening, they can alter the molecules that sometimes.
In this case, you have to know the toxicokinetics of this nitrate, then we get more information. Toxicokinetics from absorption up to the elimination process. We still have a question behind, I think.
Yeah, thank you very much.
For one people is enough? Yes. Same Anna? Okay.
Start here.
Thank you very much for a good presentation. I would like to ask you whether those crops with the high content of nitrate have a specific sign that are visible to the naked eyes so that people could detect them early.
No, you know, in a sense, we have to analyze. We cannot see with the eyes and then say, this is toxic. Well, for experienced people, it is possible. For now, I know, or you know some plants who are toxic, right?
Yes.
Yes, so we can know from literature, we can know from experienced people. I'm working on a project on a toxic plant, poisonous plant. I started this project with my student at the National Police College. So most of them are known. We have a project to identify the chemical constituent which are toxic. It is a longer study, maybe. We need some students to work on it in terms of research or thesis. And Professor Mutesa will help to get the fund, I am sure. It is a big project, but to know if the plant is toxic is by experience or by experiment.
Okay, thank you.
Thank you for your good presentation. During the presentation, you have mentioned that this kind of nitrate or nitrite poisoning and even a cyanide can have the source of the manure or the fertilizer that has been used for fertilizing this kind of sorghum plantation. What I would like to know if this kind of fertilizer or manure has not been used for fertilizing, of course, the plantation of this sorghum. We can't find this kind of those chemical elements. So I want to make a clarification on them.
I go to you.
And then provide the lethal death of this kind of toxic substances. Thank you.
Thank you very much for the question. Of course, the manure, fertilizer, all of them are the source of nitrate. And when it rains, the water will take them to the crops, and the crops can absorb it. You know, sometimes when we eat some fruit, some vegetables, you know that we can eat them with pesticide inside because of that phenomenon. So, the source for nitrate are various. I think I have a slide where you show the factors which favor the build-up of nitrate in the crops. The water, also, or the manure, the fertilizer are also the source of of building of nitrate. So it's better to use, if possible, not organic manure when you are growing those kind of crops.
Okay, thank you. We are running late a little bit. Maybe you can, when there are more questions, just direct them to Justin when time is done.
Those are my students. I think they want to ask the professor.
So thank you again.
Thank you very much. Please, we're going to talk even after. During break time, we can still discuss. Thank you very much.
So we have a little change to the program you got. So you may have seen that the session should have started with Clarisse to do her presentation, but she has 3 presentations that are on a similar topic, and she is going to combine all 3. So she now goes into the stand here and will do the lecture, 3 lectures combined into one. And we will then, after that, continue with François' lecture. So after we have been thwarted by Windows, we now try Apple. Also, Steve Jobs not delivering now.
Thank you, Oliver, for the introduction. Good morning, everyone. I'm Clarisse Musanawaganwa. I'm one of the PhD students completing my research work next month. And I'm working with Professor Leon for more than 10 years now. And I serve as a medical research analyst at Rwanda Biomedical Center. And my research work is focusing on epigenetics, on the role of epigenetics in intergenerational transmission of trauma and PTSD in genocide survivors. And we are also running different research projects, especially in genomics and epigenomics. And today my talk will be focusing on that, on the role of epigenetics in intergenerational transmission of trauma and PTSD in Rwandan population following exposure to 1994. You have to excuse us for some minutes. We have some tech issues. Why?
using their own PCs with weird troubles.
The stick is for All right, after a few technical difficulties, we now can start, finally.
So thank you very much. As I was saying, my talk will be focusing on the role of epigenetics and intergenerational effects of PTSD and associated neurocognitive outcomes in Rwandan population. This is a project being implemented by different partners. I'm having some flu, but let me move my mask. I hope I will not infect anyone. Yeah, so this project is being implemented by different partners, and the lead one is the University of Rwanda in collaboration with the University of South Florida. It's a project funded by NIH, National Institute of Health, and of course, later on we welcomed other partners like Ridge University at Brussels and Radboud University, who are the host for my PhD research. And this project has been going on, it's been more than 4 years now, and we are now at the close to the end with some interesting findings and outputs. And today I'm happy to share with you some of them for maybe future collaboration and other different networking opportunities. Oh yeah. It's going so quickly. Yeah, so this is a quick outline of the talk. Okay. Don't know. Okay. Maybe. Next, yeah. So you can do next, please. Yeah, so post-traumatic stress disorder is one of the mental health conditions which is actually triggered by being exposed to different traumatic events like genocide, like wars, famine, and other different childhood traumatic events which may trigger some symptoms like having anxiety, having different symptoms linked to PTSD. As you may see, some symptoms include nightmares, negative beliefs, aggression, intrusive symptoms, and flashbacks or avoidance. These are quick symptoms which have been linked with post-traumatic stress disorder when someone has this type of condition. Next, please. So we wanted to link this mental health concept with genetic and genomic concept, as now the literature and evidence are showing that there's high level of genetic diversity in African population. As you can see with Campbell and in 2018, in 2008 paper, they showed that there's high level of genetic diversity in African population, mainly due to genetic and culture, linguistic, and other phenotypic conditions in African population. So all these are influencing high level of diversity. Next, please. And when you look at the current data in African population, we don't have much information at epigenome level. So this has really inspired us and motivated us to look at also how we can contribute on current scope of genomic in African population, but focusing on Rwandan population, especially with the context of genocide exposure. So our project is contributing on epigenomic landscape, but exploring on the impact of genocide in Rwandan population. Next, please. So when we talk about epigenomics are these kind of studies which explore heritable changes which are inherited in the population, but which do not alter DNA sequence. But of course, they may alter or make changes in how gene function of the gene. So epigenomic studies are mainly focusing on how how changes may be inherited in human body, but they do not change the DNA sequence, but they affect the gene function of the human being. So epigenetics are having different mechanisms. As you can see, the DNA methylation is the one which is frequently studied in relation to conditions like PTSD, but there are other mechanisms like modification, like histone modification and RNA-based mechanisms. But our research is focusing on DNA methylation because it's the one which is more stable and well characterized when you want to study it in relation to trauma exposures. So again, different studies have shown different factors which are influencing those epigenetic mechanisms. As you can see, like in utero development or environmental chemicals or drugs are the main factors which are influencing those epigenetic changes in the human body. When you look at those changes, sometimes they create negative feedbacks or different health conditions like autoimmune diseases, mental health disorders in our case, or cancer conditions. So again, here, we are more interested to see how the in utero exposure during 1994 genocide, those children who were pregnant during 1994 genocide might have been affected by the exposure of their mothers, and hence maybe they created some epigenetic changes which may not have affected DNA but have been altered to create a gene function, like altered gene function, which may increase susceptibility to those mental health conditions in the future. This is our main overall goal of the project. Next, please. Next. So as I was explaining again, epigenetics mechanisms may have different influences at positive and negative level. But on positive side, those epigenetic changes may bring in some opportunities to create some therapeutics or opportunities to improve microbiomes of our human body or other different things like psychosocial interaction. But when you look at negative side, those epigenetic changes may create other different conditions like toxicity in the body or other different psychological disturbance, as I was saying. Next, please. So again, majority of studies which have been conducted were looking at psychological level, and now these days we find that stress or PTSD may be expressed or may be explained even at biological level. So this is a quick cascade also of how we think, or how scientists have been showing how trauma exposure may alter the system, the sum of the part of brain-body system. So when a person is exposed to the trauma or traumatic exposure, the hypothalamus, which is one of the brain components, tends to secrete corticotropin-releasing hormone, which is very important in triggering anterior pituitary gland to secrete also what we call adrenal corticotropin hormone, which is at the end also triggers adrenal cortex to secrete cortisol, which is an important hormone to regulate or to create balance in responding to negative feedback caused by exposure to traumatic events. So normally, when a person, next please, when a person is affected by, is exposed to stress or traumatic events, this is the, next please, come back. This is the cascade, as I was explaining, at hypothalamic level. And normally when a person is exposed to stress and is having those PTSD symptoms, he tends to have low level of cortisol, and hence he fails to control the negative feedback caused by that circumstances. So on the other side, as I was saying, the DNA methylation mechanisms, that mechanism that I was explaining before, normally the way it is, this methyl group is attached to CpG islands, to cytosine, and that attachment, that's where it starts the methylation, especially in the promoter region. So these kind of concept or theories have been proven in different studies in animal models, and even now currently they are really proving it in human population. So in our case also, we are focusing on DNA methylation, as I was explaining, because it's well stable and well characterized when you're trying to look at epigenetic changes. Next, please. So again, you can wonder how trauma can leave some legacy or marks to the next generation. When I was starting explaining, you've seen that we are interested to see how those epigenetic changes may affect generation to generation, what we call intergenerational. So normally when a person is exposed to those traumatic exposures, like genocide or war, sometimes those people, adults, they may leave some their experiences or their trauma symptoms to their next generation, to their children. So this is what we call intergenerational trauma in general. Next. So normally when we look at intergenerational process and epigenetics, when a person is exposed to those traumatic events, sometimes they tend to cause or trigger epigenetic changes in the maternal oocytes or paternal sperm. And then when that happens, that's when you say that there's intergenerational trauma. And this is what we are trying to study or explore. Next, please. So now, what is the situation in Rwanda? As you know, because of 1994 genocide, Rwanda is one of the countries which is still expressing or like observing high level or high prevalence of PTSD in our population, especially in whom we call genocide survivors. And when you look at the statistics, currently 28% of genocide survivors are having this PTSD condition. And we have 10% of our population, general population, are having depression. When you compare those statistics with the statistics at global level, you can see that Rwanda has to do a lot to see what is happening, and this is why we are posing more research questions and building different hypotheses at biological and psychological level to really try to contribute to government goals of alleviating those disabilities in our population. Next, please. So I try to make this presentation more interactive, and I try to give you a broader overview of what we are looking at. So that is the hypothesis of our project. We assume that being exposed to traumatic event in our context is genocide. This may be associated with changes in stress at biological level and at epigenetic level on different epigenetic marks, and all those may be correlated with PTSD symptoms and even neurocognitive outcomes and intergenerational effects in our population, in those targeted population. Next, please. So we have many, try to go very quickly. many sub-hypotheses. The first one, we assume that there's an association between DNA methylation and intergenerational effects of PTSD and trauma among Rwandan genocide survivors. We also assume that there's low level of cortisol level and imbalance of other important biological markers, as I've shown you before, especially glucocorticoid receptor and mineralocorticoid receptor. We believe that there is imbalance of those cortisol level in those exposed to genocide. The third sub-hypothesis also, we believe that there is a correlation of DNA methylation with some neurocognitive outcomes that we hope to test, like especially focusing on false memories. So the general objectives, we want to determine the intergenerational effects and associated biological mechanisms in Rwandan genocide survivors following exposure to 1994 genocide against the Tutsi. Specific objectives is to determine the DNA methylation status following exposure to that, to 1994, determine the level of expression of cortisol and GR and MR in that population. Also, we want to determine an association between DNA methylation and level of those biological markers, and determine intergenerational effects of PTSD and associated epigenetic changes. So there are many different now research outputs that we are having. We recently published our paper, which was exploring those DNA methylation status or leukocyte metylomic imprint in genocide survivors. That's what I meant. We were looking at those epigenetic changes in different generations. This is what we called intergenerational analysis, using epigenome-wide analysis. Next, please. So I already explained this. Next. So we did this epigenome-wide analysis based on data which were published in 2014. It's actually one of our scientists, Professor Leon. They were looking at those level of cortisol in genocide survivors, mothers and their children that they were carrying during that period. So what we did, we took the data that they generated. For them, they were looking at candidate gene, NR3C1. So we took those data, having case of 25 mothers and their children, and those who were like within country, and 25 mothers and their children, those who were outside the country, but they were matched. And we did epigenome-wide analysis, which I'm going to discuss. Next, please. So the EWAS considered only for 70 participants because of the QC which was performed to ensure that we are able to detect the changes that we wanted to see. And we used Illumina human methylation epic bit chip technology, 850K. So for this paper, we wanted to see to investigate the genocide exposure on epigenetic modification across the whole genome, focusing on genocide as the exposure of interest. And we wanted to test an association between genocide exposure and methylation level in the promoter region within differentially methylated region. So this is the overall workflow of analytical approach that we used for EWAS. As I said, we tested 70 samples using those epic Illumina methylation epic bit chip. And within those 70 samples, we returned 59 samples after doing further QC by removing, it is working? Yeah, by removing different cross-reactive probes. So with those 59 samples, what we did, we selected 5% of the probes, which were, perfect, thank you. So with these 59 samples, they survived QC for the quality that we wanted to test the epigenetic changes at DNA level. So within these 59 samples, after performing Illumina methylation analysis in the lab, we were here at USF, at the University of South Florida with Professor Derek, and we were doing this work we took those 40,936 CPGs, which were like 5% of the total CPGs that we collected. And we used them to test the differentially methylated region, as I was saying. But for those, before moving into the actual analysis, we removed the cross-reactive probes and those CPGs with low intensity and other different CPGs which were showing low intensity to not be able to detect what we wanted. So we moved on and we used methylated CPG set enrichment analysis, our package, to determine differential methylated region. This method, we used, we actually fitted the linear regression and we ranked also the CPG side, the CPG, by using Lima package, Minifa package. And then what we did, we moved on and we did the generation analysis by considering children and exposed 10 and exposed 16, in total 26. And mothers, we had total 33 mothers. And then we moved on to identify those differentially methylated regions. In mothers, we had 24. In children, we had 16 significant differentially methylated regions. Next, please. So, as you may know, the DNA methylation is checked by ranging from 0 to 1 beta values. And for our data, the DNA methylation ranged from 0 to 6, as you can see there. Come back, please. As you can see here. Next. So, as I was saying, a total of 818,720 CPG sites were retained, and only 40,936 were used to analyze those generational differentially methylated regions. So within mothers, we observed AL5C and PM20D1 genes to be differentially methylated. Whereas in children, we had TMAM204 and LDHC, which are also differentially methylated. So this is within generation analysis. And then we continue to assess now the impact of genocide exposure, which was one of our interests, the factor of interest. We associated it to different differentially methylated regions on the whole genome, and we found that BCOL, PRDM8, and VWDE are differentially methylated in all generations, in mothers and even in the children. Next, please. So these are other different genes which were detected to be differentially methylated in mothers versus non-exposed mothers. And these are a number of CPGs which were detected to be differentially methylated by genes, as you can see. Next, please. The same in children. We compared children who were exposed to 1994 genocide and children who were not exposed. And these are the genes which are detected to be differentially methylated in different CPGs. Like the first one, we had 12 CPGs which were differentially methylated in that gene. And interestingly, all those genes are linked to mental health conditions, and they have been reported to different other studies to be linked or susceptible to those mental health conditions. Next. These are common DMLs, which are well detected in mothers and children, both, which confirms that maybe intergenerational trauma might have happened in Rwandan context. So these are the key 6 differentially methylated regions which were reported in both mothers and children. Next, please. And this is how it looks like. We try to factor other different factors like maternal methylation, maternal ages, and child sex. And interestingly, we found that in utero genocide exposure remained closely associated with those differentially methylated regions detected in both generations. And you see the color intensity represents the strength of the association. We continue to measure the DNA methylation in those exposed and non-exposed group within those differentially methylated region, especially focusing on BCO, PRM8, and VWDE. And we found that all those 3 DMLs are highly methylated in exposed group compared with non-exposed group. Next, please. And then we also did t-test comparison to compare means of methylation between non-exposed and exposed mothers. And interestingly, we found that at VWDE, there is high level of methylation in those key CPGs which were detected at these genes in both mothers and children. So, in conclusion, for this first output of our project, we found that this effect of epigenetics on trauma and PTSD might be possible, and we may think about how to mitigate further changes which may increase susceptibility to those mental health conditions in our population. Second, this data, they support the theory of the effect of epigenetics and transmission of trauma and PTSD within and between generations, as you've seen. And also, there's a subset of epigenetic modification observed in Rwandan mothers who were exposed to 1994 genocide. And all these are showing potential intergenerational trauma related to epigenetic signatures. Of course, we recognize the key limitation of our study, which is the small sample size that we used for the current data, which might affect the generalizability of this data. However, next please. We are now working with NIH, National Institute of Health, as I've said, to expand the sample size and make sure that we confirm what we observed in this pilot analysis, EWAS analysis. Next, please. So, as I said, we are now doing recruitment for additional participants, and we hope to have 450 participants and again test those epigenetic changes and intergenerational trauma to confirm the findings that we observed in that pilot. So this is now the current data that we are having on this expansion, where we have around 280 participants, and we are continuing to recruit. And we assume that it's the same hypothesis that mother's exposure to trauma is associated to the child's PTSD symptoms. PTSD symptoms. And of course, we believe that those mothers' exposure and child's PTSD are moderated by mother's resilience. Next, please. So with the current findings that we are having, we find that there is an significant association of mother's exposure severity with increased child symptoms, as you can see here, in the first chart. And again, as I mentioned, here we are having like 3 generations. Mothers who were pregnant during 1994, and then those children they were having, and the children who were born after 1994. So we are having like 2 generations of offsprings. So now we are observing an association of mothers' exposure severity with an increased child's PTSD symptoms. So when we look at also severity of mother's PTSD, we find that all those depends on mother's resilience, and mother's exposure again is significantly associated with PTSD symptoms among mothers, as expected. So now we are reaching at also other interesting findings at biological level on this expansion of participants that we are recruiting. We did dosage of cortisol and MR, and interestingly, we find that after 28 years after 28 years from genocide, we still observe low level of cortisol in exposed group. Cortisol level in this group is very low compared to non-exposed group, and this is very significant, as you can see. And as I explained from the beginning, low level of cortisol is one of the symptoms or a marker which shows that a person might have some PTSD symptoms and he's not able to respond to negative feedback caused by exposure to traumatic events. So in reality, this is what is happening. Our population or genocide survivors are still having this low level of cortisol, and maybe that's why they're still having those high prevalence of PTSD compared to general population. And when you look at other key important biomarkers like mineralocorticoid receptors, we also find that there's high level of, the level of MR in exposed group is high, and the difference is very significant, as you can see. The glucocorticoid receptors, it was also high, the level of glucocorticoid receptor in exposed group, but it was not significant. And this is what expected, actually. When a person is having low level of cortisol and having one of the biomarker to be high, there's imbalance in that systems. Next, please. We continued to check this now in mothers versus exposed offspring, generation 1 and 2. And this is what we observed. The MR was very significant in exposed mothers, as expected. And the GR also was very significant, and it was very high in exposed mothers compared to offspring, and this is what we expected. However, the cortisol come back. Cortisol, there was no difference between mothers and children. We continue to observe this in, next please, next. Now to compare in exposed offspring versus non-exposed offsprings, both generation 1 and G2, and we found that the cortisol level also was very low in exposed offspring compared to non-exposed offspring, and it was very significant. The glucocorticoid receptor was also not significant in the case of offsprings. But as you can see, the cortisol, which is the main marker of interest, is very significant in this group. Next, please. So when we look at this data that we have so far, these results are comparable with the findings which were obtained in 2014. And we are still observing low level of cortisol, despite efforts which are being made by the government, different interventions which are being done to really protect these genocide survivors, we still observe low level of cortisol in an exposed group. And we believe that these findings are going to help to shape more interventions, which may help to really control these effects in these people. Next, please. So next step now, we are trying to link this data with other different data that we are collecting, H3 Africa phenotype data, and other epigenetic data that we are collecting in this new cohort that we are working on. And also we aim to do some further analysis on stress proteins, where we want to see if there are some differences in heat shock proteins 27, 70, and 90 in that population. Next, please. So, yeah, so we hope to continue also to link those biomarkers, cortisol, GR, and MR. We link those findings with epigenetics findings and psychometrics data to see if we can observe some other interesting findings that confirm our hypothesis. Next, please. So, yeah, this is, I just mentioned that. Next, please. We also want to share with you other different data that we published recently. We really did a meta-analysis work to see if the burden is still high in that population. And this is a paper that we did in 2018, I think, 2019. And next, please. It was a meta-analysis work that, as I was saying, next, please, I want to show them quickly. And we found that the PTSD prevalence, pooled prevalence in those population, genocide survivors, was very high. And these data were compared with the recent mental health survey which was conducted. You can see that PTSD is still a burden in our population. Next, please. We are also doing community engagement work where we are encouraging the community to be interactive and active in what we are doing. So we invite stakeholders, policymakers from Ministry of Health, from NGOs that really take care of those people, that community, like Avega. We invite them in the discussion. We share them the findings that we are getting to make sure that they understand what is happening. And this community engagement approach is being motivated by H3 Africa as one of the methods to reduce stigma and other different negative beliefs that are happening with use of genomic data in general. So we are using this community engagement as one of the models that we hope is going to help to reduce stigma, to also ensure that our population will be, they are secure and safe about how we are going to use their genetic data. Next, please. Next, please. Next. Yeah. So I already discussed the goals. Next, I want to finish. So as I said, the main community engagement program is being hosted by Avega, and we encourage stakeholders, we encourage policymakers, we encourage psychologists, we encourage everyone whom we think is very important in the journey of reducing or in the journey of protecting our community with mental health condition to be part of the journey of solving the problem. Next, please. Next, I want to share the lesson learned. This is our community. Here we invited them, and now they are expressing their opinions, how they think these findings are going to help them. They also we ask them to feel free to ask us or interact the way they think to help them as we move forward. Next. So we believe that community engagement is going to be an appropriate platform to understand the community when we do these genomic studies. And also, in our case, we observed strong emotional behaviors in that population, and we were expecting that. But with community engagement, it was an opportunity for us to see how to help them as we progress, and how to engage our psychologists to really help that community. Yeah. So we hope that you're going to introduce a mental health program which is going to really work with couples, new couples in that community, to make sure that we can be able to break the cycle of inheritability of that trauma and PTSD, as we'll be giving some interventions that may help them to be well and make sure that the future children, future offsprings may come without those epigenetics marks caused by their parents. So I want to conclude by acknowledging the entire research team, especially Professor Leon and his colleagues from USF and those institutions that I mentioned, without forgetting the founder of the project, the NIH and H3 Africa. So I thank you very much, and I welcome you to give some inputs or advice or any reaction on this journey. Thank you very much.
Thank you very much, Clarice. This was, of course, because I'm in genetics, it's a more interesting part for me. And I take the liberty to ask the first question. Did you somehow make sure that the stress factors, so this is, you can have a lot of stress factors affecting these genes, so if this is very specific for the genocide population, or if the non-genocide test cohort is also maybe exposed to other stress factors that could inflict the same amount of methylation? Yeah,
thank you very much, Oliver, for a good question. Yeah, so when we were recruiting, first we introduced some psychological measures to really first test the condition of the status of our cohort, where we first tested if they even have symptoms of PTSD before recruiting them, both control and cohort. But also we had some other psychological measures which control further environmental stressors in both groups. And then in addition, with the analysis that we are making, we make sure that we do sub-analysis by taking into consideration individual exposures and individuals' conditions. Yeah, when we do the analysis, we make sure that we do sub-analysis based on control exposures and group cases exposures, yeah.
Okay, thank you. More questions? Yep.
So thank you very much for a presentation that honestly is humbling in its scope and its potential impact. So really amazing work. I find your findings really compelling, identifying specific genes that may be impacted and maybe the mechanism of transgenerational trauma. Another way of approaching kind of broad epigenetic impact that our group has been exploring is the impact on biological aging. And there's some really nice tools to estimate using the same data. Overall accelerated biological aging, and I would be fascinated to see how that accelerated aging translates down the generations. I would suspect, based on your data, that we see accelerated aging from the outset, that already offspring already start their lives with kind of an advanced age, which we know is linked to worse health outcomes later in life. So it's, if you haven't considered it, I
definitely
would be fascinated to see what you find. But yeah, thank you very much.
Of course, I think it's, we can start from there. And, yeah, those tools are from, they are R-based or, yeah, thank you very much, Brad, yeah.
maybe to add on that, because we thank you very much, Clarice, for a nice presentation, by the way. So to add on that, we also have a biobank with different specimens. So using, we're also having RNA samples we can use for transcriptomic studies in that way. Even can go far for functional studies, as you mentioned. There are very interesting candidate genes that we need to do further, some functional studies. And then to come back to Ori's question, so this is really very interesting. Not only this genocide event, so we have been recently contacted by researchers from Ukraine. What is happening in Ukraine, like a war? This is also an impact for this. And then they are really interested to see how the population who are being impacted by this war can also be studied. Some of the, to see if we find some of the epigenetic defect that she mentioned here.
Thanks, Prof.
Maybe when you go to Ukraine, they can also have some samples before the war, after the war, if you have some blood bank information.
Yeah.
So, more questions, of course.
Thank you very much for very interesting presentation. Yeah, but there is a research that has carried out on mother and the children with a sample of 25 in the country and outside of the country. So my question is, does location or environment can affect the change of genes or the stress in terms of intergenerational traumas? Yeah, thank you.
So you want to confirm if trauma, the environment can affect changes in genes? Definitely, that's where I started explaining. It depends on those environmental exposures that you're saying, but many studies were conducted in the context of war, in the context of famine. So they really confirmed that environmental exposure may affect epigenetics changes. But as I said, epigenetic changes does not mean that you are changing the DNA. Instead, they change the gene functions. And those gene functions may be changed and maybe bring negative behaviors or negative consequences, which may lead to different mental health conditions or any medical conditions. Yeah.
I think he was more referring to if you have the second group that was outside of the country, if they maybe have the different diet or exposed to different toxic substances like the smoke or something like this, if these effects can have the same effect and then for this PTSD stress in the other group.
That I cannot confirm, but of course, based on the hypothesis, they may have different effects, and depending on the level of exposure. That's how I think. Yeah.
Usually, just to add on this, usually if you have, you will have the same or different amount. If you have a significant difference between the two groups, and the other group is, but in group it's still more or less one group is significantly different, but inside the group it's the same. Then you can most usually you can exclude other factors for this, because they will not all have the same exposure to maybe a diet or to smoking or to alcohol or whatever inside the group. It will be the different difference inside the group than between the two groups. So there's should be it's a self-regulating problem.
Yeah, thanks, Oliver. Just to add on that, so as you've seen, the level of cortisol, for example, was very low in exposed group, those that we call case group, compared to those we call control, who were outside the country. So there's already a difference. And at epigenetic level, as I said, there are different effects and different factors, as you said, smoking and other different factors, taking medical treatment and other different factors. This, we hope to control them in these large sample sets that we are collecting, and we are collecting those data on alcohol taking, smoking, health conditions. We are collecting different information based on H3 Africa phenotype tools, and we hope we are going to control all those factors and see if there are changes or differences among the group. Yeah, that's the next steps, as I was saying, yeah.
Okay. Thank you for a wonderful presentation. My name is Andrew Ivan. I'm a basic neuroscientist. My question, I just need more clarification. When you took the baseline data, we know, and it has been established that like in teratogenesis, where we have the primary transmission or alteration, either DNA or epigenetics can occur. So the clarification I need is that we know that the genocide took place, that is, it was very intense in 1 month during that 1990s. Is it maybe 1 month or 3 months, perhaps? And we know that transmission occur within the first trimester, if there is anything that can happen to the next generation. I just want to know, are we saying that the epigenetics that occur, does it occur as a result of continuous environmental information, or does it occur due to mutation? That is, mutation of people that were affected during genocide? Because we have study that shows that if you traumatize a laboratory rat and a pregnant rat, there is tendency of stressors that can be transmitted to the offspring. So I want more clarification to know, within these 3 months of intensive genocide, and we have a lot of intergenerational transmission, does this intergenerational transmission started before the 1994? Or was it during the 1994? Or was it after the 1994 with this every year Quibuka discussion? So I just want more clarification, because all those factors I mentioned, they could also be a continuous stressor to alter the gene, the epigenetics. So that is what I wanted to know, or to see how this, your wonderful study, can help either to continue discussing it or to stop discussing it so that we can reduce the stressor or things like that. Thank you very much.
Thank you very much, Ivan. That's a very interesting question. Very interesting question. Yeah, for clarification, for us, the hypothesis, we were assuming that those children or babies that they were carrying during that period, in 1994 genocide, might have, that intense exposure might have affected epigenetic values for those in maternal oocytes, which might maybe have been transmitted to those children during that period. But of course, we do not exclude that concept that this continuous exposure or continuous intergenerational process. And of course, we believe that as we progress, there are other environmental stressors that are maybe contributing to those changes. But what you observe is that we still observe the difference among the control and cases. If you've seen that all of them, well, are still alive and we match them, as I was saying, but we still observe that difference in exposed group. We do not know if it's a continuous exposure or if it is that change that happened during that period of 3 months during 1994 genocide. Maybe those are other research questions that we may explore.
Clarice,
Again, we do not.
To compliment
Yes,
on
please,
that.
Prof.
This
Yes,
is a really, this is a really interesting question. So that's how even we started this project in 2012. Actually, the intense period of 3 months was one of the reference for our study. But we know that the genocide even started before. Yeah? When you see people have been exposed for that period before, and then when we got a chance just to have among our sample size, some of those mothers who were pregnant during 1994, who were also traumatized before. So you could just see that intergenerational process happening before. But of course, what is interesting is this strong scientific evidence, which shows that those mothers might have different methylated regions versus others who have never been traumatized during that period. So you can also make another, like copy paste with another event, which is not genocide, like the war. I was just discussing the Ukraine. So if you see an event happening just at that time, and then it is a continuous and very intense, producing the level of methylation to be high, so maybe the intergeneration process will start that date. So it's really a continuous process, but different depending on which population being targeted and the date that population have been just exposed to that event. Yeah, this is really very interesting. It was also our goal to get this NIH grants, to explain how we can just explore the intergenerational process within the generations.
Thanks, Prof. Yeah, thanks, Prof. Leo. Yeah.
All right, do we have more questions or comments on this nice talk? There's one. Let's take this to the last one and then we go to the break.
Yes.
Oh, there's one too.
Yeah,
okay,
thank you very
those
much.
two.
Yeah, thank you very much. I'm Dr. Agustin from clinical psychology. I have some comments, not questions. I start by a joke. When former president of Ibuka went to study in one of the country of Europe, his university, one of the lecturer told him, you as a genocide survivor, it will be difficult for you to succeed because you are traumatized. So the best way or the best thing to do is to go back at home in order to seek healing. Yeah, that joke is I call it a joke, but it's not a joke related to what we have to consider in terms of when we are conducting research related to what history has called ethnic group. What I am emphasizing, we have to consider, to take, to be aware of mental representations related to other factors. People can say, as you are a genocide survivor, automatically you are traumatized. So I don't say that the study done has been conducted in the same way with those mental representation. We congratulate you of what you have done, but I would like to ask you if cultural beliefs, cultural consideration, mental representation of your participants must be considered. Other thing, I would like to ask you if we can say that study is completed as soon as it has only regarded the one social group, why not it will be completed when it will be also extended to other societies or social groups like Jewish, like Armenians, who have been experienced also genocide. That's why I would like to have your comments related. Can we say our study, is it 100 completed as soon as we don't extend our study to see how trauma can be transmitted? Because genocide, if we regard as genocide, it has been prepared considering the same methodology, and it has the same effect. Thank you.
Thank you, Augustine. Thank you for a good comment. What I will just add on is that there are many studies, same studies, which were conducted on Jewish population, and some references were provided, and maybe I may be happy to discuss further for your clarification. And again, for social studies looking at social beliefs, I would like to inform you that there are some in-country scientists, Professor Vincent is here, and others who are looking on that component of social beliefs on the impact of genocide. Of course, we'll be happy to collaborate. For us, we are more focused on biology and genetics, but we are happy that I can see Professor Vincent here, yourself, Ivan, and this is an opportunity for networking and making sure that we are informing the public. Yeah, thank you.
Thank you. My name is Wimana Ive. I would like to ask this question because it's related with that you are speaking about the literature. Of course, you have done the literature about the other side that this study have done. For example, there are places like Jewish people, they have suffered like how we have suffered in Rwanda. So we have seen that there is a problem in mental health for the people in Rwanda more than other places around the world. So, but I would like to know from your discussion after your findings on the markers, on that markers that have been changed, how can you compare the severity of these changes of changes of DNA compared with the studies that they have conducted on the other side of the world? Thank you. And if you have found what's the recommendation for that side that we can learn from them for us if we find such same situation? Thank you.
Yeah, I will give maybe a quick example of those Holocaust population, Jewish, as you were saying. They found like the same epigenetic changes, but for them, they reported more on NR3C1 gene, and they were more focusing on candidate genes. But maybe the added value of our data is that for us, we looked at epigenome level, and so, and the other thing is that they introduced many social interventions. And even in the US, I think, with our new collaborators, Ahmad, they were having some proposal to have some nutrients which may be helping to control those levels of stress. And we were hoping to introduce those kind of intervention in our group to see if there might be some changes in the future. Yeah. Thank you very much. Yeah.
Thank you. I know there are a lot of more questions directed to you, but I would like to invite you to discuss this during the break. We'll now have our coffee break for a quarter of an hour, and yeah, feel free to discuss any topics from this session again then. So you see you in 15 minutes.
Thank you very much. Thank you, Oliver.
François Xavier and his topic during DNA sampling during legal abortions. Yes, it's coming. Just getting ready.
So welcome back from the break. This is the topic I'm going to discuss with you. It's not a findings from a research as maybe previous presenters did, but for me it's more a concern that I would like to raise and then ask from you some contributions to improve this sector. So I ask you to pay attention so that you can contribute and help us to improve this issue related with the DNA sampling during illegal abortion. Those are what we are going to discuss, and during our discussions, this is some definitions. I don't really want to take much time on it, but particularly what we call abortion is any end of pregnancy without consideration of the time of age. But in the medical view, we consider the viability, and this viability differ from country to other countries. But in Rwandan context, in Rwandan law, termination within legal abortion is limited with the circumstances which are defined by Rwandan law. I will show you a slide showing what are circumstances where abortion is allowed or there is exemption from criminal liability, because according to Rwandan law, you know, abortion is still illegal. And DNA, I am not really an expert on DNA. Me, I am working in the field of forensic medicine at Kachiru Hospital, especially in Isange One Stop Center, and I worked more than 10 years in forensic medicine, and sometimes I combine because my field of study is forensic medicine. Just show you that what we would like to focus on is this part of DNA to help to link the perpetrator and the victim, because we are in the field of sexual violence. And this will be considered as an individual evidence, or what we can call also these characteristics evidence, which is different from class evidence, because here we link. This is, we determine. We don't need an amount of evidence to give a high probability, but we can really link the person, the victim, and the perpetrator using this DNA. And then around abortion, this is what I promised you to present. This abortion is still illegal, but from 2012, in the penal code, there was an exemption, and at that time, only 4 conditions were there, and then the victim should present a court order in hospital or to the medical doctor. But now, during from 2012 to 2018, only 10 cases along the country was registered of people requesting safe abortion. So in 2018, you changed the law, and then we had another one, law on infraction, and this one gives more details on conditions allowed for abortion, and then the court order was no longer a requirement. So this is what was the new with this law. The court order was no longer a requirement. And those conditions, they are there. Those are 5 conditions. If you become pregnant because of child defilement, if you are pregnant because of rape, if you become pregnant because of forced marriage, if you become pregnant because of incest up to 2nd degree, or if the pregnancy jeopardizes the health of the mother or the fetus. So on those 5 conditions, you can consult any hospital, and then they can proceed for safe abortion. And the ministerial order give more details about what we can do within the Rwandan framework. And the remaining problem was that all cases, majority of cases that are asking for safe abortion, they filing under investigation. And as you may know, some of you were there, according to this ministerial order, in the hospital, we don't ask for evidence. So if a female person of 25 years come in the hospital and tell me that I am raped, I am not asked to ask the person what are the evidences that you can present to me so that you are maybe victim of rape. I proceed without asking proof of the acts of violence. So, and for child, majority because they are in the child defilement, those cases, they filing under investigation, and we need further evidence to handle the case. And if the person is pregnant, sometimes even the history of how the child was defiled, you don't have any evidence. The only remaining evidence should be taken within on the fetus or during the abortion. And the problem so far we have is that from Rwanda Forensic Laboratory, from the Ministry of Health, from the Rwanda Investigation Bureau, we didn't have any guidance, any protocol about what we can do to proceed in the correction of evidence and even on the channel of transportation, even if we can follow for transportation other channels. But for correction, for taking sample in our country today, we don't have any protocol or even guidance on it. This is why I decided to present this in this forum of forensic scientists to help us on how we can develop it and even to give some ideas on what this can be done. So this was the objective, these discussions of what you can do. But what is in the literature review? Now, what I'm going to present is not a findings from a study. A study will be maybe conducted after. Now, this is what is from the literature review. According to what we can find in the literature, some specialists of DNA who are there or others who have more knowledge on it, theoretically, you can have the DNA from the amniotic fluid. You can have it from the plasma, those cell-free fetal DNA. But the question is, does our laboratory able to perform the tests of DNA with those components, those fluids, etc.? You can have it from placenta. So, and the problem will be for placenta, and if is an early pregnancy, what can we do? Sometimes we are advised to ask the person to keep the pregnancy until a certain level of age, but asking someone to keep the pregnancy while he wants maybe abortion is also a problem, because the law allow her to undergo this abortion, and we as health providers, we want to ask the person to undergo with the pregnancy at a certain age, which is not fair on my view. And also some tissues, the problem with the tissues is from storage and transportation. Because having tissues from Sofa, from Kigali, like Rusizi, like I don't know, transportation, storage, etc., will be absolutely a problem, because they should be bring those samples to Rwanda Forensic Laboratory for examination. And what's there, it can be taken by swabbing, either swabbing of red muscles, swabbing of some other organs, swabbing from products of conception, swabbing from products of manual vacuum aspiration, and even sometimes they speak about even swabbing on flowing blood during abortion. But for this, we know that if this is flowing blood, majority of cases, you will have the mother DNA than the fetal DNA. And also, for tissue and cells, you can have this unfixed placenta, you put it in normal saline. You can have some tissues, they will be put in normal saline also. You can take bones, but for bones, according to literature, not only it's very difficult to make this analysis, but also there is zero quantity. And for plasma, the problem will be how we will proceed if we don't have equipment to proceed. So those are some what we can find, what we can do. So theoretically, swabbing is very simple, because after swabbing you dry and the transportation will be easy. And tissue with normal saline, the transportation, it will not be really easy. I think now this is like the last slide, because giving you the floor and receiving your the feedback from you, your advice, your input, to help us to improve this field related to DNA sampling on abortion. So, what is the current practice? Because even if there is no protocol, even if there is no guideline, even if we don't have any reference, but we use what we found in literature, and what we do now, we swab red muscles. This is because some medical doctors, they called me asking, what can we do, what can we do so that we can swab, because the Rwanda Investigation Bureau is asking us to swab for DNA. The advice so far, I advise them to swab on red muscles from thigh part, and then dry and pack, and after send it to Rwanda Forensic Lab. But sometimes also it works, and on other times they packed after direct swabbing without even drying, and it becomes a problem with the contamination, with moisture, with etc., etc. So this is the advice we gave, and then now, as I told you from the beginning of this presentation, I am not going to discuss because I didn't conduct any research, but some recommendation from you and some advice from you will help us to improve this sector, because this is the statement, abortion is it's like a right for the person. Correcting of evidence will be a requirement from Rwanda Investigation Bureau, but health providers who are in the field, because you know majority of abortion will not be conducted at the RIB, it will not be conducted at an office of police or any other office. It will be conducted in maternity. So this will still like a problem, and we want from you to receive some advice to improve this practice related to DNA sampling during illegal abortion. I thank you.
Thank you very much, Dr. Francois. Any question, welcome. Please, you can ask your question or any comment.
Thank you for the nice talk. Just last week I got two DNA from 2 abortions for DNA analysis to get a paternity test. And one was a fresh one stored in isopropanol, and the other one was fixed in formalin, but very shortly. So you can take more or less everything. You can even take the bones because they are so small and brittle, and just you can dry them out and just crush them a little bit, and it's still working for DNA, no problem. And you can take any other of the tissues for DNA, should all work. So because if it's a fresh abortion and this is stored not too long in formalin, it's very valid for DNA.
So you are advising to take samples from tissues?
You don't have to take a specific tissue. You can just take. So we used some muscle and skin from the thorax, and we used some thigh bone just to test it out if it's working, and some rib, and was all working very well. You can just insert the ribs even into the extraction buffer, and it usually has slightly acidic, so you get will dissolve a little bit, and then you get all the DNA you want from bones from fetuses, and from even if you get further back in just a few weeks or for a few days old fetuses.
Okay, this is a contribution. Do you have an idea of the age of pregnancy that can allow to do this sampling?
This would be valid for every, so it doesn't depend if it's in the first days of a pregnancy. Of course, you have to differentiate the tissue from the mother's tissue. Mostly if you have only a few days, they will not find out that she's pregnant, so it will be aborted without any knowledge. But if she knows she's pregnant and there is only abortion, they have some abortion material going off, or it's even done by some outside other countries or something, so more or less legally outside of Rwanda, then it's no problem to take the sample. The doctors can take the sample, put it in alcohol, ethanol, or isopropanol, and store it for a while. Or when you need it, you just can take it out, dry it, and use it for the DNA extraction.
Okay. And what about the swabbing of red muscles? If we have this experience?
You can swab the muscle, you can use the muscle in total, so it doesn't really, it depends on how big the fetus is. If you want to save some time, you can do the swabbing, but because the swab is easier to process, but you can also use some cut out muscle tissue and just use that.
Thank you.
Any other question?
Yeah.
Thank you very much. It is a very good topic. Matthias Renge, the Dean, School of Faculty of Health Sciences, Uganda Matters University. In Uganda, abortion is illegal, and there are a lot of social and economic factors that are contributing to that when someone does an abortion, unless there is a reason beyond reasonable doubt that you can terminate the pregnancy. What I've learned is that personally, when I'm doing ultrasound at one point, no mother comes to the facility to say, I want to know whether my baby has congenital abnormalities. They always want to know the gender or the sex of the baby. They just want to know the gestation age. And it is always disorganizing them at the end of it when they deliver a baby with a congenital abnormality. So unless we try at a certain level that DNA sampling is done, and maybe we try to examine before that, and we get the key markers and we confirm that the child is likely to have unbearable congenital abnormalities, that's when they can terminate. But this one should be done by the physician who is trained, and it is not done even by one physician. It is cascade of health workers who will agree that yes, this baby will not survive afterwards. But you realize that because of the social aspect of life, they wouldn't wish to terminate someone's pregnancy until maybe, and some of them give chance and say, let us wait and see. Because when you stop them from doing the abortion, the only effect that is going to come out is that they will resort to unsafe abortion. And that's why some countries, they try as much as possible to do genetic screening before, and then endeavor to see that these babies whom they are producing are free from unbearable congenital abnormalities. Thank you.
Okay, thank you for your contribution. So meanwhile, you don't have any experience about DNA sampling in Uganda on products of abortion? Yeah? Okay.
Thank you once again. I think what I, as an anatomist, let me put it that way, where our duty is to localize structure and positioning, either isolating, preserving, or transporting them to forensic laboratory. In terms of early analysis, there's what we call amniocentesis, where within 3 weeks of gestation, the expertise of a radiologist and a gynecologist, where a sample can be taken, the amniotic fluid can be taken through a simple process called amniocentesis. And there's also what we call tissue aspiration, where the placenta cell, a small piece of it can be aspirated and then fixed in PBS, there's phosphate buffer solution. And then where you can do all type of analysis to know even the genotype, any neuronal abnormality, morphological abnormality, histological, everything you can from that, and you see the amniotic fluid, and then the placenta tissue cells. Then with the help of a PBS, phosphate buffer solution, to help determine if we can accept, if this pregnancy can be allowed to proceed or not. So it has to do with a trio, a help of a radiologist, a help of a gynecologist, and then an obstetrician. So those are the 3 key principal people that are supposed to help so that we can take a decision to avoid unsafe abortion. So that is my input, I can say. It's just a comment on that part.
Okay, thank you for your contribution. And because there may be what is needed is, there is another question there. It's related with legal abortion. So as you may imagine, there is no need to go with amniocentesis, because even the whole products will be coming out. Maybe these can be used if we wanted to keep the pregnancy. But those conditions, the person need abortion. So you don't need really to go inside for maybe, because you will at the end terminate and you will have all those things. You see, sometimes we use this, the procedure if we wanted to keep the pregnancy, and then we would like to see if it's a pregnancy that can be terminated or that can be kept inside.
Thank you. You presented about the law that established how you are allowed to make a legal abortion. So we know sampling many times is for further investigation or investigative purpose. So you told us that if someone comes and want to abort, a medical practitioner does not ask why and if that rape or other have happened. So why sampling? So what is the use of that sample? Thank you.
Thank you very much. This is an interesting question. If you can see among conditions, there is also the condition related with a child pregnant or case of child defilement. So this is according to other Rwandan law related with the child protection. If the person pregnant is a child, they should know who is the perpetrator of the person. So this can apply if it's a case of rape, meanwhile someone aged of 18 and above. But if you are under 18, this is a criminal case and it should be investigated by Rwandan Investigation Bureau. For other cases, forced marriage and incest, etc., they don't ask for any other evidence. But for child defilement, you know, they should. The investigating agents should know who is the perpetrator. This is the reason for why even this DNA sampling, even if it can be applied for the person, the raped person, if you are under investigation, but also for child defilement, it can be applied to all cases of child defilement. And majority of person demanding abortion, this legal abortion in hospitals, they are child. So more than 8% of people demanding abortion, they are child. You know, I presented to you that we have so far, we have more than 2,000 cases, and more than 1,500 they are child.
So we take one more question.
Thank you very much. I'm Dr. Frijanse, and I would like to ask, is it regular to do like DNA sampling in every minor child who have been pregnant? Like, is it allowed to do sampling? The other thing that I'm asking, so as usually we are doing like swabbing. So like, I need to know if for you to bring this issue, is it because swabbing is not like accurate as the other method, or there is another thing behind it? Thank you.
Can you repeat the second question so that I can give maybe?
The second question I'm asking, normally we are doing swabbing, even for the cases of abortion when it is requested, or for the rape. So I need to know if, for bringing this issue, if swabbing is not accurate.
Okay, thank you for your contribution. For the first one, for all cases under investigation, if you proceed as a medical doctor with legal abortion, you should take sample because there is no any other remaining sample that can help the judiciary to link the perpetrator and the victim. So it's mandatory. But if maybe the case is not under investigation, if the pregnancy come from another child, because it's also possible, if the perpetrator is not known or maybe is really missing, the case is not under investigation, the sampling of DNA is not really mandatory. So, and why do we bring this issue while we were using swabbing? But where are you, why do you use swabbing? This is one of my questions. Do you have any guidance? Do you have any protocol? Do you have, where do you found, why did you use swabbing? Maybe you use it because it's from colleague, maybe you use it because it's from, I don't know, but we don't have any protocol. So it's not because swabbing is not relevant, as maybe one of the DNA experts gave us the advice. Swabbing is possible, tissue is possible. There are many techniques that can be used for sampling to analysis, but what we would like to have is to have one common understanding of where to swab, when to swab, how to store, how to make maybe transportation from the time you are taking your sample to the time it will be analyzed. Because we don't have any protocol, even if sometimes after swabbing, I don't know if you dry or not dry. So this is why this subject was presented here, because we are in the forensic forum where we can have a lot of ideas from experts in this field and have something which is relevant and which can help us to have accurate results from what we took. We have one expert from Rwanda Forensic Lab. He can give the experience. Sometimes some samples we took from our samples, they don't found anything, not because they are nothing during correction, but because the techniques, how you take it and how you manage, you handle the evidence was not proper. He's there, and I have, I had a presentation with the National Prosecution the last week about this concern. 100 sample, and among them, 40% you don't find anything. So you lose money, you lose equipment, you lose reagents, you lose many things because you don't have any protocol. This is one of the reasons why I am presenting this subject, just like awareness, and then put together our effort and produce something which can help and enhance the level of our evidence. Thank you.
Thank you very much, Dr. Francois. As you know, in Forensic Summer School, we come from beauty to another beauty. That's why we're going to call the next presenter is Fred. Please welcome.
Thank you so much, Dr. Albert. My name is Fred, as he said. So this topic is not on this, but it is number 6 in the abstract. So I'm very happy to be here again after epigenetics and post-traumatic stress disorder, and then to abortion, legal and illegal abortion. So I'm going to discuss about my master's thesis in international criminal law. So in 2020, when no one was allowed to fly, everyone was in house, I decided to go in law because I'm an investigator. So I pursue Masters in International Criminal Law at the University of Rare Adventist of Kigali. So the topic, the thesis title is Use of Forensic Evidence in Investigation and the Prosecution of International Criminal Proceedings. And I take ICC as a case study. So I will talk a little bit about ICC and the other International Criminal Tribunal, but my main focus will be on how they collect evidence, how they present them in those international criminal courts. So some pathologists here, you may ask yourself, why am I need to know this? It's because as we see, the world is coming together and there are different atrocities have been committed around the world. For example, Ukraine and Russia war, we had some case in Eastern DRC. So all those places need scientists like me and you to go there and investigate crimes. So as I've said, there are many atrocities. So we need forensic scientists to do an investigation so that we may not heavily rely on testimonies in criminal tribunal. So historically, this is the aim. My aim is I will discuss a little bit about international criminal law in history, how it has developed. And then I will not go through the whole thesis because it is around 80 pages, but I will just take some cases where these forensic evidences have been used or was used, and some cases that failed because of neglecting this evidence, the scientific evidence in prosecution. So, and I will just show some challenges still available in international criminal justice. So historically, from 1945, after Second World War, the international community decided to establish international criminal court to try some military tribunals. This is Nuremberg Military Tribunal, established in 1949 in Germany. And they established it to punish those who committed atrocities during the Second World War. So they even established the Far East Tokyo Military Tribunal. Later in 1993, another court was established. It was established by the United Nations Security Council, which is ICTY, to punish or to prosecute the offenders of atrocities in former Yugoslavia. So it was adopted by Resolution 827, and its headquarters was in Hague. So later, the Rwanda International Criminal Tribunal for Rwanda was established by United Nations Security Council by Resolution 955. The headquarters was in Arusha. I think everyone here in this country know about that. To prosecute the offenders of genocide against Tutsi in Rwanda. So soon, in 1998, the countries get together to say that why can we always based on these ad hoc tribunals? Why can't we have permanent courts? That's how the International Criminal Court came up and started its operation in 2002. Its headquarters also in Hague. It was adopted by Rome Treaty in 1998. So this International Criminal Tribunal is the one that I have focused on. Because to establish an international criminal court, first of all, you need to think of statute and also the rule of evidence and procedure. The rule of evidence and procedure guides how the evidences are collected and how they are used. So so international crimes, this ICC, its Articles 6, 7, and 8 stipulates about the international crimes. By now they are mainly 4. Crime of genocide, crime against humanity, and war crimes, and even crime of aggression. So currently, up to now, the ICC has prosecuted, the prosecutor of ICC has sent an indictment of 31 cases, but I have taken my time to go through all those indictments to see where these forensic evidences were used, how do they correct evidences, and even I go to the statute and rule of evidence and procedure to see how do they correct evidences from crime scene to the court. So, so basically, the investigation at the domestic level and international level, they are almost the same, but some differences. Here, the RIB is responsible for conducting investigation, but at the international level, the prosecutor, office of prosecutor, has that duty according to rule of evidence and procedure in all courts, ICTY, ICTR, and the ICC. So even the mandates, while in our domestic laws, the criminal law and criminal procedure are the ones that guide us when we are conducting investigation, how to conduct investigation, how to correct evidences. So, so I use documents. I review the cases of the ICC as I have discussed. So here is the case I have selected. Because I could not go in all cases, I have decided to use this case is a case in ICTY. So I can tell you why I have decided to use this case is because it is one of the successful forensic-based investigation since the 1949, whereby the investigators, the office of prosecutor, they invited the forensic pathologist, forensic anthropologist, and the other fields in forensic to come and assist them in investigating this case. So this man was a general in former Yugoslavia, and they committed atrocities in 1993. So this grave was found in 2005, and there were many bodies buried in, it was mass grave. So they invited the scientists. So for him, he was claiming that we did not commit, it is not a war, it is not a war crime, it is not an international crime, but this grave has soldiers, they were combatants. So after excavating the grave, they found that the registers on the victims, they were not soldiers. They examined the clothes, they found that those people in the grave were not were not combatants, were civilians. Some were medical doctors, others were children. So the court concluded, based on those evidences, the trial chamber of the ICTY concluded that this man, General, has committed an international crime and then prosecuted. So this also, those investigators also helped to relocate by examining DNA of the remainings to document and to profile the victims, send the dead bodies back to, send the remainings to their owner. That is the humanitarian act of returning the victims to their families. So another case here in Rwanda, it is a case against Georgi Rutaganda in Ictr. So the prosecutor were basing on the testimonies from here that this guy has committed genocide. But this reporter, Nick Hag, recorded during 1994 genocide, he had a record of this person killing a daughter. So from that video, they concluded that this Rutaganda has, the court prosecuted him against committing, they prosecuted him based on this video record, the footages that were taken by this reporter. So one of the failed cases I have taken from many that I have examined, I have analyzed, is about Katanga and Jongori Chui. They are from Democratic Republic of Congo. So in pre-trial chamber of the ICC, they have decided that these people were innocent, and because there is no sufficient evidence to establish the guilty to them. But later, in 2012, they attended the crime scene. They do scene reconstructions, and they examine the grounds where the things were being committed, and then in chamber, when the prosecutor appealed, and chamber concluded that he is convicted and sentenced of 14 years of imprisonment. So after making my research and all the cases I have examined, such one from ICC, I have observed that the reason behind the failure or the failing to use the forensic evidence in international criminal courts, one is insufficient legal guidance of evidence admissibility, because Article 64 of the ICC statute stipulates that the evidence admissibility has to be be determined by the court. So another thing is administrative and lack of logistics, because they don't have sufficient amount of money and other things, so that can also be a problem. So those two, they affect and they lead to unnecessary complications and prolongations of the cases in the International Criminal Court. So as conclusion, I have seen that there is a need of a reform by establishing the specific legal procedures to be obeyed by International Criminal Court in forensic evidence examination and forensic and even chain of custody of evidence from the scene to the court and so on. So I have recommended that those international bodies and even international community at whole can work on that. Thank you so much. Any question?
There's a quick question, one or two. Okay.
Thank you, Mr. Fred. I have seen you, of course it is a master's thesis, but I would like to know whether so far there is a study, maybe from your side, because I know where you are working, where there is a study, is there any study where you can determine where forensic evidence have been used in courts or in other criminal proceedings? Thank you.
You mean, sorry, you mean
I mean another study, another research where forensic evidence have been used in the criminal proceedings.
Yeah.
I mean a research, not just by saying. I know that it is being done, but we don't know how much, because I myself, as someone working on a forensic lab, what we do produce forensic evidence, but we don't know how the forensic evidence impact in the criminal proceedings. Is there any research so far about that? Thank you.
Yeah, thank you. Yeah, as I said, I did not go in all, but they are there. There are researches made, and I did not go in the whole development, how they have been developed since the beginning, but there are very many researches made. Even here in Rwanda, Dr. Thierry Murangira did it on how the forensic evidence impact in criminal justice. We have also other international journals on how the forensic evidence have been used in the courts.
Thank you. Let's give a big round of applause to Fred. So we don't have much time. I would appreciate if maybe Kagame during lunch, they can reach out to you for more questions. So we're going to watch a small video on a presentation by Wala Rebecca, who is is a clinical psychologist and psychoanalyst, and he has been working on supporting people who have faced rape and abuse.
Good morning, everyone. Actually, good afternoon and greetings from Brooklyn, New York. My name is Viola Rebecca, and I am a psychoanalyst who was born in Poland. Currently, I am living and working in New York City. I'm a psychoanalyst and a researcher, and my research and my work is dedicated to the war rape survivors. So maybe it would be really important to just warn everyone who is able to attend this presentation about trigger warning. So if you experienced previously any kind of sexual violence or other form of trauma, be aware like what I will share with you will affect you, maybe will affect you. So please make decision if you're able and you're willing to expose yourself to the uneasy topic such as sexual violence during the war, trauma related to the sexual violence, long-term consequences, PTSD, and war rape survivor syndrome. I was born in Poland after the Second War. My family has a Jewish roots. Most of my family were killed during the Second War. A few of them survived. One of them were my grandmother, who spent over 2 years in Nazist camp in North Germany, Ravensbrück, a camp dedicated to the women. She was subjected to the experiments, and also she was gang raped during the liberation. She experienced oppression from the Nazis during Second War, and also she experienced trauma related to sexual violence during the liberation from the liberator, because she was gang raped by the Russian soldiers. This experience deeply affected minimum 3 generations in my family. My mother, she was born after the Second War. She was exposed to the trauma of my grandmother. However, during this time, nobody spoke, nobody tried to even understand sexual violence as traumatic events. No one was willing to work with survivors of sexual violence. So this trauma passed next generation. So that's the reason, major and private reason, why I become a psychoanalyst and why I started working many years ago with survivors of sexual violence. First with survivors of domestic violence, survivors of any kind of regular violence in Poland, and then I started working on the field, working with international NGOs, working in refugee camps, mostly in many different places in Africa. So I was able to visit Rwanda a few times. I used to do some research. I used to work with mental health providers in the country. Last time I went to Rwanda in 2018 when I decided to conduct more research regarding survivors of experience of survivors of sexual violence from the genocide in 1994. Hannah Arendt, a Jewish American journalist, brought to the world attention a completely different understanding of cruelty. She was the first journalist who openly reported the trial of Adolf Eichmann, one of the Nazis' famous bureaucrats, on April 11, 1960 in Israel. She wrote an outstanding piece, Banality of Evil, which contributed to expanding the social narrative regarding complex trauma at the time. Her narratives inspire me to think deeper on how to change general diagnosis, treatment, and allocation of resources for trauma survivors, especially those suffering from the war rape survivor syndrome. War rape survivor syndrome comprises many symptoms, physical and emotional, and unique experience endured by women who have survived being raped during war and other conflict zones. As a humans, we have an innate desire to know and understand, and that's normal. We need to identify and articulate our pain so as to start the healing process. Words are powerful symbols that can be add significance and relevance. Through words, we can share experiences, feelings, thoughts, and trauma. An event or experience that is overwhelming and painful must be first identified and conveyed in order to begin the healing process. This identification must emotionally additionally take place between understandable and visible social, political, and emotional cultural context. This context is essential in trying to understand war rape survivors' experiences. War rape survivors are mostly invisible to members of their communities and homes, even when they live with relatives who themselves have been subjected to war-related trauma or nature are constructed by the life drive to experience war or to abandon of war rape is against our human life drive power. To counter war and rape is to face total destruction. Both elements destroy families, communities, nations, and the survivor's personal sense of safety and identity. It can be common for survivors immediately after their rape to deny pain and downplay the magnitude of their massive trauma. My work with war rape survivors has brought for me another level of understanding of the complex trauma meanings. This work particularly opened my eyes to the indescribable aspect of the human experience and moved my attention to the lack of individual narratives of war rape survivors and their subsequent experiences. So, because I was able to travel, because I was able to work with survivors in many different places, I decided to collect their story. I decided to move forward with understanding of long-term consequences related to the post-traumatic stress disorder. Currently in the United States, we are approaching another one level of the description of the long-term consequences related to the Ptsd, and we calling this complex post-traumatic stress disorder related to the events such as war rape experiences during any kind of war. So because of my interest and because of time what I was spending in many different places in Africa, also in Rwanda, I was able to work with survivors, particularly from Rwanda Society. Last year when I went to Rwanda, 2018, I was able to conduct additional 68 interviews with survivors of sexual violence during the Rwanda genocide. Last year also, I got an opportunity to publish my first book, Rape, A History of Shame, Diary of the Survivors, dedicated to the survivors of sexual violence in many different war zones, but mostly survivors of genocide in Rwanda. So following understanding of work with survivors first, I was doing some research, and I came across to the study published and dedicated to the long-term effects of conflict-related sexual violence experiences at the end of the Second War. And this study compared 27 elderly who went through with sexual violence at the end of the Second War with other people who they were exposed to the war trauma, however they didn't went through with sexual violence. They were using modified version of the post-traumatic diagnostic scale, and they assessed trauma characteristic and post-traumatic stress disorder symptoms, and they were using brief symptoms inventory to assess current psychopathology. Additionally, they were using measure of post-traumatic growth and social acknowledgement as a trauma survivor. Social acknowledgement questionnaire were used to assess two mediating variable in post-traumatic condition of war rape. Women exposed to the conflict-related sexual violence reported greater severity of the PTSD related to the avoidance and hyperarousal symptoms, as well as anxiety compared with female long-term survivors of non-sexual Second War trauma. And there was majority showing 80% of these women also reported severe sexual problem during their lifetimes, to only 19% of women who experienced no sexual war trauma regarding their sexual problems. Women exposed to conflict-related sexual violence also report greater post-traumatic growth, but less social acknowledgement as a trauma survivors compared to the survivors of no sexual war trauma. That is extremely important topic because many of the survivors of sexual violence during the Rwanda genocide showing and seeing and sharing like they are invisible for the society. So during my research, conducting interview, collecting story, working with survivors with their trauma, I decided to create a new understanding of long-term consequences for the survivors to describe their experiences. And I'm calling this war rape survivor syndrome related to the re-victimization process, what is affecting trauma related to sexual violence, and is also related to the social ostracism, social lack of the support, social shaming and blaming, what is the major problem for many of survivors who are trying to resolve their issue after sexual violence during the war, especially right now. I'm referring to the experience during the Rwanda genocide. So as we know, Rwanda genocide is one of the most painful and most bloody experience in the human history. We know already like over 1 million people were killed, probably approximately 500,000 people were sexually assaulted, raped, tortured. And we are talking about, right now after 20 years after genocide in Rwanda, we are talking about already long-term consequences related to the sexual violence. We are talking right now about the long-term psychological and social consequences of unresolved issue, and that need to be addressed, that need to be named, that need to be brought to the clinical attention to work through this unresolved problem to avoid epigenetics, meanings like long-term consequences passed to the next generation after trauma in 1994, unresolved sexual violence, and current research showing the findings related to this research, like unresolved sexual violence during any war, any genocide, is the major reason of passing trauma to the next generation. So seems like is extremely important right now to address this issue and work through with survivors of sexual violence in 1994. Also work on stigma related to sexual violence, work on language. If I'm correct, in Kinyarwanda, unfortunately, it's really difficult to describe really straightforward experience such as a rape, because rape translate to Kinyarwanda as a word doesn't exist. That's what survivors, what I was able to interview, share with me. Like in Kinyarwanda language, rape as a word doesn't exist. Meaning of rape, description of situation of the context existing, but this particular word still is hidden and invisible for the society. As I said from the beginning, to start healing process, we start using appropriate language to describe our experience. I hope I will be able to share with all of you more information about my work, particularly dedicated to the war survivor syndrome. Thank you for your attention. Enjoy rest of your day. Best regards.
Thank you, Viol, for the presentation. So if you have any question for Viol, please reach out to me and then we can give you the email and you can write to her directly. Now, the second speaker is myself, so I will introduce myself. A bit of, as they're pulling up the PowerPoint, so I have, I think when I was here, I know some of, maybe we have, it was a journey when I was first as a student, now I'm a practitioner, and now I'm also a teacher. I'm teaching at the University of Rwanda and the University of Melbourne, and I'm doing some research. By the research I'm focusing on now is also a practice-based research that is a bit lagging behind in our work, especially from mental health. If you can see a number of colleagues were presenting some of the case studies, so I have now developed more of an interest in the cases that I receive at the clinic. How can I be able to move forward and be able to document it and make some publications? So this is the recent paper that was published that I'm sharing. And over the practice, when you look at the history of mental health in Rwanda, just to quickly catch you up, we see that pre-colonial in 1900, there were no systems in terms of structures for mental health as specialized services. So there was no like a psychiatric hospital or anything. So the way support was done for people who had either schizophrenia, who had any mental health related difficulty or problem, was through the traditional practice. I think like any other medicine as well. But as we go to colonial period, we see an introduction of a structured and a systematic way of responding. And around, I think in around this time, that's when there was no psychiatric hospital, but people with mental health difficulties or who had mental health problems would be taken to the prison. So this prison initially wasn't built for being prisoners, but it was built to host people who had mental health problems. So now we are looking at getting from the community, now going to a psychiatric arrangement, which is the system. So we're seeing this introduction, and the first psychiatric hospital being established in 1972, which is the Netherlands Neuropsychiatric Hospital. The reason why I'm saying this, when I'm talking about stigma that we always sing, and I think it has become a trend, that is a mental health stigma, people don't accept services, don't go to services, you need to also think it through a historical lens, not only as a scientist, but also as someone who is investigative as a researcher. So the recent survey that was done, it showed that only 5% of people who had mental health challenges consulted mental health institutions, despite them being decentralized. I think it correlates or resonates with what Clarissa was mentioning in the morning, that care can be there, treatment can be there, support can be there, but people moving from their homes to those psychiatric institutions or to those psychiatric services requires something else. And that's where now we come in again as mental health practitioners, but also as a society, not only the practitioners. So the challenge has been, how can a government, of all that they have said, the effects with the transgenerational trauma, with effects from the rape, the abuse, and all those services that are being provided for mental health support, how can we have people reach out? So actually, this is a fun fact. For everyone who wants to make a consultation, he calls, hello, is this Joseph? And be like, yeah, I wanted to meet you. And be like, meet me for what? Oh, sorry. Meet me for what? They always, oh, I wanted to meet you. I will tell it when we meet. Because there's that kind of tense of it. And sometimes, if you told someone, I don't know if anyone here, if I told you that in the morning I was at Ndera, what would you think? For the equivalence of Ugandan is Tibutabika. So if I told you I was at Tibutabika, what would you think? Yeah, go on, sir. Yeah, but you're putting it in a nicer way of a patient. People think you're mad, you're crazy. What was wrong? And I have had some friends who say, oh, I can't even buy a plot in Ndera, right? Because the association. So that all has an impact on the behavior of how our families or families of your loved ones, how they seek support. So my interest now was, how can we be able to go back to the community and put care there? So there is a, let me call it, nickname it as Project Hope, and this is what we're going to be trying to see. How can we honor and earth the local knowledges and survival wisdoms of the different people who have expressed, so call them like ex-service users, or people who have used psychiatric services, or people who have used mental health care. How can we be able to honor that? And one of a Brazilian psychiatrist, who is now a community psychiatrist, mentioned that exclusion and diminishment of people's local knowledge is a key contributor to psychic misery. And when people's local knowledge is devalued, this leads to interiorization of misery. Then misery becomes an interior experience. So people, when we don't give honor, or when we don't value the ways of which the community, you saw from the colonial approach of care, so when we don't give value to the collective nature of the communities, of how they respond, and be able to bring as researchers or practitioners something that that aligns with how they have been doing things, then we face resistance. So that's what the psychiatric approach is, and that's what was my curiosity in this research. So one of the things was, how can we draw inspiration from other collective responses to mental health, including those that are supporting people who have faced sexual abuse. This is in Zimbabwe, where the work of some of the care counselors in and some of practitioners in Uganda who have written some of the work in terms of raising our heads above the clouds, honoring those community. So this is a group of Promame members. They are members who have been consulting at Indera Neuropsychiatric Hospital, and now they have formed support groups that they meet every other time. And each group has a minimum of 20 members, and they have formed structures. So I wanted to understand what are the ways of which they have been responding to that stigma that was happening to them, and how have they been able to adhere to some of, build a resistance and a solidarity. So in doing that, I was in acknowledgement that as I'm joining, they know you're joining as a researcher or a psychologist or a practitioner. How are you going to ensure that there is safety, but also they are going to be comfortable with you being around, because normally those are closed ones. So that's why I initiated, this is a metaphor, but I took, for those who watch football, you know red cards, a red and yellow card. So they became the referees, that they could give me a red card and a yellow card in terms when I was collecting data and trying to ask them that if they felt the question made them feel overwhelmed or feel stressed or they didn't feel like responding to it, for Rwandans, you know, they are not going to say, oh, I'm not fine, I'm not okay with that question. They will just keep quiet. So how can they be able to demonstrate that they are not okay with it? They could give me a red card. So whenever they gave me a red card, I could stop from pushing to that question. You know, you're dealing with the sensitive. So this is something that I was very curious. How can they feel safe as they are meeting? Then the other thing that also what Vio was mentioning is the language. If I go speaking about stigma, what's stigma? If you ask a local community member stigma, what's that? What's depression? What's schizophrenia? So how can you be able to deconstruct the language and have people speak in a way that they feel resonates with what they feeling? So we are trying now to see how can they best describe stigma in their own ways. And that's when some of the metaphors, like Akato Imungu, started coming up. And then we are no longer talking about stigma in the conversation, but now we're talking about Akato Imungu, which they have very much knowledge about. For all, I don't know if you all know Imungu. Do you know it? Imungu, na yivugaga mu Cyongereza. It's Imungu. So it's the cowpea that affects maize. So how has this affected them? They could demonstrate it in a way of the cowpea, how the cowpea infests crops. And they were so much knowledgeable that they could speak the different categories of how Imungu affects. For example, I would learn that from the stories that the cowpea, the way it affects the northern part of Rwanda is not the way it affects the eastern part of Rwanda because of the temperatures. And that, when you correlate it to stigma, the stigma doesn't affect people the same way. It affects people differently, depending on the social support, depending on the cultural context, and depending on how people are supported. So these are some of the rescued words and phrases from the, I won't read it because of that. And then we are trying to create what was called, we call communitas. It's a, how can we invoke collectiveness, but retaining identity of each individual? As here, all as a group, how can each of us feel as a group, but every voice that is individually is also respected? My identity is not lost in a group. That's what we call the creating communities. So drawing from that story, we are able to develop a richer one, that in which we are able to talk about things that are sensitive, but also close to people. Then developing metaphors work well with Rwandans. They like to speak Mumigani, Munsigamigani, and all that. So when you engage in that, then you're able to go into meaningful conversation. Then, what were some of the ways they were responding to the stigma, the collective ways? Some of it was staying focused to their dreams and values. That if they knew what they wanted as a group, and they knew what they wanted to attain, despite the harassment from the families, they could build upon that. Then, for example, what Yvonne mentions that, that because they expect you to do wrong, even when you do something good, they feel that it's illness making you do this thing. So you know that if I told you now that I have, let's say bipolar, or I have schizophrenia, or and that has been prescribed by the doctor, that everything I'm going to do might be thought that I'm doing it because I'm not stable, right? So how do you stay focused when people think you're not stable? So the values became something meaningful. Keeping silence. For some of the members, and this was a bit of contradictory to the common trending thing of break the silence. Always people say, oh, can you speak? Speak out, then you'll be healed. You will be okay. No, it doesn't work that way. So for some of them, felt that keeping silence was a way of responding and taking time for themselves before they engage into a conflictual relationship. So these are kind of a discourse to what is commonly known. Some of them, this is another thing that we need to understand from the African, the culture, and mostly the African context, that when we are talking about that, people need also to be economically empowered. So they have engaged, when they meet in such groups and engage in community activity, then they are able to reclaim their life within the neighboring societies. So financial independence became a way of restoring and reclaiming their life. I don't know if that makes sense. But poverty, actually, and this is a fact that is not recorded, but poverty in Africa or in our countries, and even now, people are starting, scientists and other social scientists are starting to look at poverty as an illness itself because of what it causes or what it leads to. I don't know if it makes sense. So people who are, whenever they are deprived of resources, then they are able to they are not able to gain their independence. So then there was speaking self. I won't delay on this. Then some would engage in the songs. I had made here a short one that they were playing, and the other thing that came out was that some people think mental health is a relative to their own social. So they think it's bewitching, it's something that. So I went with some, I wanted to prove this. So here there was a group of some Australians who traveled, and they were also having struggled with mental health, one that have been. And then when they went in the community and they saw, I'll use this, but for our German colleagues, so it's a Muzungu, so the white person. So when they saw the white person that they were also struggling with mental health, they were like, oh, we thought it was only a local problem. By that sharing of knowledge, then it builds opening. I think you can understand that from the context. So I just wanted maybe as we pause off that when we approach any community or mental health challenge, always as a researcher, have an open eye to honor or respect what is happening. And currently, we were privileged to have a small grant with me and Herbert and some other colleagues to have some trainings for medical officers in terms of forensic science. But what I play in that role, or what we play from the mental health perspective, is how can even the medical officers that are opening up bodies in the autopsy, that are dealing with cases of GBV, suicide and others, be also supported. So that's another thing we are looking at. And what we are building now is, what we are looking at, how are some of experiences, for example, the across doctors, how each can contribute in strengthening, because when a young graduate medical doctor is given a role to play in a hospital without clear support in terms of well-being, then it affects their work. So I won't go in detail because my timer is out. So if there's a quick question, I would take one. Otherwise, we can go to the next presenter. Thank you. Okay, so one question, then what we're going to do, we're going to pause for lunch, and then we come back for the other sessions. We have a bit of, yeah.
Yeah, thank you very much, Carissa, Joseph. Only a simple question related to the comparison between imungu and stigma effect. By definition, I don't know if you got time to define imungu. Imungu in Kinyarwanda is defined as simple insect that affect sorghum or beans when they are in store, and also they are affected by those small insects. So I would like to know if someone who, or you, who in comparison with those imungu and stigma, the comparison is related to effect by, yeah.
Yeah, maybe because of that I didn't go in detail on it. So the explanation to it, or the correlation, is that the way imungu affects crops is how stigma affects their lives, the people who have experienced mental health. So it affects that it eats them inside out, and there were also comparisons. So there is that imungu that affects inside, and there is that comes from outside. And which one was more dangerous? For them it was from inside. So the way it affects, and the parameters, the conditions that favor imungu to grow in crops vis-a-vis the conditions that favor stigma to grow within the people who have experienced mental health. So that's the notion. The reason is when we are talking imungu was much easier and closer because they have so much knowledge about imungu than speaking about stigma. So stigma became abstract, whereas imungu became an experience near of the conditions they were experiencing. Okay.
As a stigma is related, or it can be heard by social work. If you try to reduce stigma, it's better to incite people to join others and to incite others to join or to collaborate with. So imungu is very, very deep. I was thinking about irungu, not imungu. Thank you.
That's another opening.
Last question? Okay, thank you very much. My question is, how did you, okay, the people that you focused, the communities that you focused on, because we have in Uganda, there are some slum areas and some regions of the country like Karamoja, where we have people that have been put in a bracket of mentally disturbed people, yet it is just the environment, like poverty. People are living in a life of psychiatric people, yet it is just poverty. So which people did you focus on when you were doing this?
Yeah, so the focus was people who have been admitted in the psychiatric hospital, and now they are released into the community. Yeah, so those are the ones that I was focusing on that are now in the community. Yeah. Okay, so thank you very much. Let's go for lunch. They gave you the directions. We'll try to be quick, 45 minutes as well, and then we go to the next session. Thank you. Bon appétit. Please stand up all. So we're going to the next session. Before we do that, I want us to do a small warm-up because it's going to be a long hours. So let's all stand. Just a second. So you're going to count with me. So you count 1, 2, 3, 4, 5, and then 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, and then we'll go down until 1. Did you get it? And you're going to do it with energy, right? So you're going to shout like 1, 2, 3 quickly, right? So we go. Are you ready? So I'm going to count 1, 2, 3, and then we go, right? 1, 2, 3. Let's go. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4. 1, 2, 3, 4. I must be a horrible teacher. Okay, so let's go. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2. 1, 2. 1, 2. 1, 2. Quickly. 1, 1, 1, 1. Okay, so the next session, as you can, we're going to hear from Kagame. He's going to take on the two presentations together. So the first is the use of forensic biometrics in a criminal investigation in Rwanda, and he will combine it together with fire, arson, and arson scene investigation techniques and challenges that he did with Byukusenge Janvier. So you're welcome, Kagame.
Thank you, Carissa, for welcoming me, and thank you also for an exercise. It was not easy afternoon. So coming back, and this is my last time in Forensic Summer School 2022. So I'm so thankful for the organizers to give me this opportunity to make this presentation. I'm going to discuss about use of forensic biometrics in Rwanda. Rwanda perspective. So later I had another with Byukusenge Janvier, but he did not manage to attend this year. So he suggested that I can do that presentation related to the fire and the arson crime scene investigations. So so this is the scope. Introduction, aim, problem statement, review, biometrics in Rwanda, then up to recommendation. So according to researchgate.net, biometrics refers to the measurements and statistical analysis of people's unique physical and behavioral characteristics. It means it's studying the physical and even behavioral characteristics of an individual. So in forensics, we do it for the purpose of investigation. They can be used to identify for victim identification or even suspect identification, so that they can be used by investigators to administer justice. So forensic biometrics is now available here in Rwanda. We do have some, as I have discussed yesterday, we have database of fingerprints that now currently we use Rwanda Investigation Bureau, we use the database from National Identification Agents, Identity Agents, and we also, I have already discussed about this, identification of suspect, of the victim, and even documentation. So in Rwanda, investigation, according to our criminal procedure and even Rwanda civil and criminal procedure, investigation is in the hands of RIB, except some cases whereby police can do investigation, but they don't have the power to send the indictment or to transfer the case to the NPPA without authorization from RIB. It means when police conduct investigation, they do bring that investigation, that case to RIB, so that we can transfer the case to the National Public Prosecution. So except that there are some cases which are conducted by the military when the cases are not. Thank you. So when cases are involving a military personnel, that time we don't investigate. The military auditorium and prosecution, they are the one to conduct it. So basically, this is the chain of forensic evidence in Rwanda. So this is LIB. We can do request analysis from Rwanda Forensic Lab, and this is NPPA. When we get the feedback from Rwanda Forensic Lab, we send them to NPPA, then to court. And even court, they have the judicial power of making some request of evidences to the RIFR. So currently, there are three, according to Interpol, there are three main used in international identification biometrics. One is fingerprint technology, whereby there are fingerprint database that we are used to identify the suspect or even unknown dead body, victims, and so on. We have also DNA technology, whereby there is the DNA profiling of individuals, and then you keep the DNA for future, then in future can be used to identify either suspect or victim. There is also facial and voice recognition. But in Rwanda, currently we have only two. We have fingerprint, AFIS, and we also have the ones I have mentioned in National Identity Agents. So this picture represents this is national ID before this kind of technology appear, and this one is the new one that we have now. So every Rwandan who has a national ID, we can easily get his or her fingerprints. So when we suspect someone to be a criminal, we can use this, or even identification of unknown dead bodies. We use the fingerprints, then we go for national identity agents to check whether we can get to know that person. But there are even other biometric data that now Rwanda Forensic Lab are developing, which is AFIS, Automated Fingerprint Identification System. So, so this is Rwanda perspective. I have already mentioned about it, and these are legal frameworks. So we had no laws, but now the government have adopted laws, different laws, and established institutions which are in charge of the biometric data management and information. For example, we have this law, Preventing and Punishing Cybercrimes of 2018. We have law of 2021 relating to protection of personal data and privacy. We have law of 2016 governing information and communication, and we have some institutions too. This one is Cyber Security Authority, established in 2017, and we also have Rwanda Information Society Agency. So now, when we came up with a case in one investigation bureau that someone is not known, we have what we call fingerprint identification kit, is this one. So we use them we take fingerprint, we have a system that can take fingerprint, then we go for analysis. So this is it. Last summer school, we discussed about this and how it works. So even if we have those that is in progress, we have some challenges still, like infrastructure, they are there, but not well developed, and we don't have DNA and facial identification recognition database. So we conclude with that research revealed universal development of forensic biometrics method applied worldwide, like DNA identification, fingerprint identification, and facial recognition. And we have seen that there is a progress in Rwanda in matters related to forensic biometrics and its use in crime scene or criminal investigation. But there are also some challenges, as we have discussed. So we recommend that it can be good for us because criminals, they are almost everywhere. They are changing, their things are changing. Someone can be in Kenya and commit a crime in Rwanda. So we suggested that if possible, there can be a center for biometrics in Africa or even in East Africa particular, so that it can be easier for us to exchange information with the other jurisdictions. So thank you so much. If any question or comments? So he's suggesting that I can do one I have on fire investigation with Janvier, because he's not here, then we can have the questions together. The first presentation, the next. So fire and arson scene investigation techniques and challenges. We are going to see the techniques. So now everything you have here, this building, this mic, everything here can cause fire anytime. As we are developing, every part of the world can face the challenge of fire. If you remember, some years back, something happened, explosive in Beirut, Lebanon. So everyone can be exposed in fire. We used to face challenges here of fire. So there are some techniques that we use. They are the ones I'm going to share with you when we are at the crime scenes. And there are also some challenges that we face because there is a difference between fire caused accidental fire and even the arson. So first of all, before I go here, arson is, according to our criminal law that providing offenses and penalties in general, it defines arson as deliberate set fire to one's property or your own property. So fire investigation is enormous and costs society around the world. It is almost everywhere, as I have said. So there are things, we do investigation, and it plays a pivotal role in solving cases. When we attend crime scenes of fire, it helps to know that we do investigate to know if the fire was deliberate or was not deliberate. So this is a legal principle, nurum crimen nurum poena sine lege. If there is no crime, if there is no law, no crime, and there is no penalty. So currently here, in Rwanda, Rwanda Investigation Bureau is the one which is in charge of
conducting all criminal investigations. And some times we work with fire brigade from police, because you cannot go to investigate the fire while fire is. We went there after the fire has been extinguished. So we enter at the crime scene, but before we go there, there must be some fire brigade personnel. So Rwanda Investigation Bureau. After that, this is statistics, how the fire is a problem, no need of going in. How 1.3 billion fire reported in United States in 2015 and so on. These are data. So the reason of presentation, as I have discussed, is to explain the fire awareness, then different techniques and some challenges. Then fire investigation. When we go at the crime scene, what we need to know, we are investigators, we want to know what is the cause of the fire. The cause. So to know the cause, you need to know the origin of fire. So I will go back to it soon, the components of fire. It is somewhere. So normally, fire is made up of three things. When you stop one, you manage the fire. One is fuel, another is oxygen, another one is heat. So those three, when they come together, of course, there must be fire. So to extinguish fire, you must remove one. And most cases, the fire brigade, they used to stop oxygen because it is easier to manage, and even sometimes you can reduce. If it's like this building, you can remove these chairs. Fuel, it's not gas or something else. It's anything that can be burnt. So this is how fire develops. Normally, for this building to be burnt doesn't need huge amount of ignition. Just small can cause all this room to fall down. So there are three stages. Mainly is growth phase, whereby fire develops, and it reaches at the stage where it is no longer developing. That is called fully developed or stationary phase. And there is decay. When things is here, if you can see this image, here fire starts, but here this is the end. So after everything is burnt, it stops. Or if something is missing, like maybe if you stop fuel, oxygen, or heat, one of it, so it starts to decay. So fire triangle, I have discussed about it. Yeah, these are key terms. The origin of fire is where the fire began, and the development of fire, I have discussed or read about it, is how the fire grows. Of course, development depends upon ventilation. If there is oxygen, if there's space where the fire can access oxygen, it can be easily spilled or grow. And even the volume of searing height, location of the fire, and even the condition, if it's in or out, or affects the development. So here are the techniques. At first, when we are at the crime scene, the reason why we want to know the source or the origin of fire is that from that particular point, you can identify the cause of fire. If it's a fuel, if they have exposed some heat there, then after that, we take sample from the remainings there. Then that remainings we transfer them to Rwanda Forensic Laboratory for further chemical examination to identify really what is in this. If it's like, if it's fuel, or if it's something like can cause to be burnt. So after that, these are procedures, normal procedures of crime scene, attendance. You need to make sure that, because where there is a fire, you must be safe. You secure yourself by wearing PPE, personal protective equipment, and you even check whether there is nothing that can damage either personnel or yourself. So you also identify witnesses at the crime scene. if at the first time there were witnesses, those can give you some reading information that fire has originated from this place to that place. So another thing is external and internal examination. It's like crime scene search. So another thing is documentation. You need to do some photographs at the crime scene. You know what was inside. How did you get at the how do you find the crime scene? What we call PV de constat, and you also do photographs. You video record. You do sketches, and so on. Then you do establish origin of fire, as I have discussed, and the cause of fire. So then evidence collected, you transfer it to the RAB, Rwanda Forensic Lab. Then at the Rwanda Forensic Lab, they can use gas chromatography techniques or even depending on the quality or quantity of the evidence, they decide which method of identification or identification techniques. So this is fire debris analysis. As I have discussed about them already, chromatography, spectrum, those are the lab. And then there are some challenges, as I have mentioned. So our purpose of investigation, we do investigation because there might be some claim. There is a suspect. If there is a suspect, there is a crime. So if you have a crime, if there is a crime, it means that you are going to prove, you investigator, that this fire was arsonist or was maybe accidental. So after getting those informations and even laboratory report, you can conclude that this fire was set deliberately or was not set, was accidentally. So challenges is that sometimes those arsonists, they are very intelligent enough to hide the evidences. For example, there is a case that we went there. The owner of the company was making a loss. So he decided that, so because I'm making a loss and I can burn my house, and then I claim that something happened here. So that is a fraud. That can be part of motivation of committing crimes. So they have motivation. So when they do it, they intend to hide the evidences enough. So it costs a lot of time. Another thing, it's hard. You can get even all those things that you need, but to prove the relationship between crime scene and the suspect, it's not something which is just easy. That is one of the challenges, and so we recommend that there must be some fire detectors. Currently, of course, we refer to the lab, but our mobile, we don't have these fire detectors. And we again recommend that there can be trainings on the investigations of fire scene investigation. We don't have a lot about this fire scene investigation. Most of the time, police, because they are firefighters, they are the ones who take trainings. But investigators, we don't have specialists in fire investigation currently in Rwanda, so that we can say that one is a specialist in fire investigation, and we don't have even school where you can go to study these things. Only modules at the university or even at school. So there is a need of training programs. Yeah, that's it. Thank you so much. If any question or comment.
Quick question?
Thank you very much, Fred Kagame, for the wonderful presentation. As we were presenting, what came in my mind is when we have fire brigade stationed in a certain area of the country, probably in the center, and I imagine you getting a call from a customer or a owner of the building that it is now burning. By the time you leave a certain part of the area to go to the other side with our traffic jam, as concerned individuals, what are you having in place to see that you can expedite the process of making sure that the fire is put at a stopover by probably quickly reaching that particular area, like using aeroplanes? Is there any such thing that we can save a lot of time and save people's property and lives? Thank you.
Yeah, thank you so much for a good question. So currently here we have those cars of fire brigade, but it's lucky that here in Rwanda we have about 10, so in legions. So, but some years back we used to have like only one, working countrywide. So sometimes people of the community, they used to use the blankets, so sometimes they don't get facilitations from the police, but now we have them. So that stage of an airplane, I don't know now. Yeah. So maybe I can go to recommendation. So another thing you reminded me that I did not discuss as a challenge. Sometimes you go at the fire scene, and because of fire brigade, they use those big. So they do destroy the evidences. You came, you find a lot of water at the ground. You cannot see anything, like you take like sample. You can't even identify the source, the origin, and it's a challenge. Yeah. Thank you.
I don't think we can hear you. You have to.
Yeah, maybe it's just to complete what Mr. Kagame is saying about fire, because we have also like a policy on prevention. The first one, it's one he mentioned about not having a fire brigade based only in Kigali, but having some other base in the province. This one. The second one is related with the inspection of some buildings to fulfill some requirement, and having, if they have those measures to intervene while they are waiting for cars to come. And the third intervention is the intervention conducted by the fire brigade at the national level of sensitizing population on how they can manage some kind of fire which sometimes don't even necessitate the intervention of the national level, of how they can intervene, how they can use blanket, how they can use muchanga. This is, I don't know, in, yeah. And those are kind of sensitization which are conducted along the country, and even with the village level to intervene first at the level while they are waiting for the national or the local intervention from fire brigade. Thank you.
So I'm going, please try to.
What I can add on is now there is a campaign that is being made by Fire Brigade, Arendt, Rwanda National Police, whereby they are reaching out to local communities, training them how they can be their own firefighters instead of waiting for the police to come. So if you have like fire extinguisher on your door or your window, you need to learn how to remove the pin, you can stop this fire at the beginning instead of waiting for it to be developed. Thank you.
You had one question? No? Okay. Is it answered? Okay. It's answered, luckily.
Yes, you can ask.
Thank you. My name is Mignon Murirgwa, and I'm a student from University of Rwanda. I have these two burning questions I want to ask. My first question is regarding forensic autopsy. Does it always require consent? If the owner someone's dead and their relatives doesn't really want the body to get involved in forensic autopsy, is it mandatory that? Okay, I want you to explain more about the consent, and when the relatives don't really agree with the autopsy, what can be done? My second question, I'm going to address this since we're in College of Medicine and Health Sciences, about these medical errors made at hospital, but the preventable ones. These medical errors made either by miscommunication, bad prescription of medications that can lead to death of a patient. Do these medical errors also taken as a clinical matter when making investigation? Do these doctors or team doctors that made these medical errors a preventable one also taken as criminals? Thank you.
Thank you so much for your questions. I will try to answer your question, and I have Dr. Xavier here will help me to clarify more. For the first question, the consent of the family members depends upon the case. In Rwanda criminal law, we take crimes as against society, not committed against an individual. So that's why even RIB is there to do investigations for the case, because if you cannot punish, it means you are creating the society of criminals. So the consent in case of autopsy comes only if it is not a crime. So if it's a crime, there is no need of consent. That time they do autopsy. I don't know, maybe doctor will help me a lot on that. And your second question is about medical malpractice. So we have medical doctors here who do investigation on medical malpractice. They can clarify much about that.
Okay, thank you. I think one of the first question was already answered. We have, according to Rwandan law, we can classify the first. We have like 3 types of autopsy: the forensic autopsy, the hospital or clinical autopsy, and then scientific autopsy. So, for scientific, which is conducted just for scientific or research purposes, you need the consent from the family. For hospital or clinical autopsy, if after maybe giving some medications you are hospitalized, but medical doctors, they are not really comfort with the diagnosis, and they want to go further after death to know why. So this is a clinical one. They ask for consent. If you don't consent, the autopsy is not conducted. But for medico-legal autopsies, because of the purpose of investigation, even if the family is not don't want it, it will be conducted because of the force of law. This is for the first question. For the second one, this is the current practice. In some countries, even for medical legal autopsies, they require consent. But in Rwandan context, we don't. It's not a requirement. We don't even contact you for consent before. We only explain you what we are going to do, and we try our best to handle the body so that you found it in a natural way. But you can't oppose yourself to the autopsy if is a medical legal one. For medical malpractice, this is one of it's like a growing issue within our country because we are developing in many sectors. From 2012, we have a specific law on it, and even it was proposed to have some committees and subcommittees at the level of provincial and level of district to help during investigation and to receive some alleged cases of medical malpractice from people from community, etc. So what are we doing to prevent? One of the prevention in view of justice is to punish those who are responsible. So it started, and we have a national committee at the Rwanda Medical Dental Council. The committee is in charge of investigating and giving the recommendation to the court, and we have a memorandum of understanding with the Rwanda Investigation Bureau for a joint investigation, because sometimes medical malpractice is an issue of medical, and a lawyer investigator don't get really, if you go in deep with the file or with the prescription, with it, you don't understand what is there. So we have committees at the national level who, which are in charge of investigating those medical legal errors or medical legal malpractice in general. This is one of the level of prevention. The second level of prevention is related with, because sometimes a medical doctor is a person like any other person. So it's to improve the training, because if you improve the training of the person, you are preventing some errors. This is the third level of prevention, and even of solving the problem is related with a new teaching in medical schools with the issue of ethics and solving medical legal issues. It's now new. The student in the last year, this year, they have the course, and it will be done in all other, every year. It's now in the curriculum. So this is new, to teach people how to communicate with the patient, how to solve any issues arising from patient and the treating doctor, and even within the community and family members. So I think, I don't know if it was the concern about medical legal, but according to what I get from the question, this is in general what I can give you as an answer. I am one of the members of the National Committee, which is in charge of investigating and solving issues related with medical malpractice in Rwanda. Thank you.
Thank you very much for that deep response. I think it's a hot debate even out there, so during break you can reach out and continue if you have more clarifications about that. Thank you. Can we give a round of applause for Kagame? Thank you. So the next, Ashimirwe? And he's going to be talking about individuals' handwriting variations under the influence of alcohol. Okay. I wish we also do the typing.
Thank you very much. I'm called Shimirwe Japhet.
I'm by names, and I'm a Chief Inspector of Police by rank, and I'm a forensic specialist in Rwanda Forensic Laboratory. Currently, I'm working as a DNA specialist, but I have a very big background in question document. That's why today I'll be talking about handwriting. So for the handwriting, we should try to understand that the handwriting are made by someone, but due to some influences, the writer may change due to the motricity of the what I can call neuromuscular motricity. So if I can go with my presentation, I will start by saying that there are some tools that are used, mainly the handheld microscope, when you are doing the analysis. This study was conducted in the state of Gujarat in India. And again, what we were trying to focus to understand what was the influence of alcohol on someone's handwriting. That was the aim. The aim was we were trying to understand, of course, the influence of alcohol, and then we were trying to establish whether there are some criteria that should be used while trying to identify the writers. And for methodologies, we used a sample of 50 handwriting, and this is a very small sample in number because we were in a dry state. Actually in India, when you are in a dry state, those are the states where you don't drink, and yet we're talking about alcohol. So it was not easy to get a big number of samples. Then we were supposed to be having like 5 other subjects that were supposed to help us to conduct the subjective analysis. They were not part of the team, but you should give them the samples and try to give their views. And again, we should take the samples. Someone should write before and after taking after consumption of alcohol, and we should be using the neat whiskey to make sure that we have the accurate intoxication for our participants. And then to calculate we're using the blood alcohol content, our calculator, just to calculate how much the participants were drunk, just to be able to write. Those are some of the materials that we used. And then coming to the result, just to be quick, we observed there are some observations that we made that in general, about 60%, they were changing their handwritings, while like 47%, they should change the alignment of the handwriting. Because you understand, when someone is not sober, they will not be able to write. They will be just changing the alignment of their handwriting. I think we'll go, just to be quicker, I will go through these numbers. In general, 40%, they did not change their handwriting. They resisted because they knew what we were doing. And regarding the size of the handwriting should change after consumption of alcohol. Those are the numbers. And then when it comes to slant, the way the slant of the handwriting should change. But you'll see that because there's these things of, in forensic science, we do understand that when someone is used to give a given handwriting, they will not change because this is something that will be burnt, that will be in their mind on how they form the letters and whatever. Those are the changes in connecting stroke, where we have like 93% did not change. And we see even the tremors that will appear. For people, we'll be having like 53% will be having tremors in their handwriting, while other 47 will be having no tremors. And again, if we go to the general pictorial appearance of a handwriting, we see that like 10 out of 50 did not, they changed completely their handwriting, while the other 40, they were changing their handwriting under the influence of alcohol. To be quick as possible, from our result, we found that it could be concluded that alcohol, it is a central nervous system depressant, and it affects the motricity, someone's motor skills. So therefore, the handwriting of a person under the influence of alcohol shows some deviations from the original handwriting. So as I said before, there were some limitations when we were trying to study this, because I told you that it was in Gujarat, which is a dry state, no beer, no liquor, so it was not easy to find the participant, because most of the people there, they don't drink, so it was not easy to find the participants. So in this case, in the future, we are planning to do this here in Rwanda, where we can be so many drinkers so that we can find more participants. And again, as I said, the sample size was not adequate enough just to draw conclusions. And again, the tolerance level, because most of the participants were not tolerant to alcohol. So it was not easy just to intoxicate them as much as possible as to give a big output. And in the future, we'll work on the increased sample size, and we think that before we collect samples, we don't have to notify the participant what we are going to do. And again, apart from handwriting, we are planning to do the signatures as well. And then we also think that we may study other states, other state conditions, like for handwriting and signatures for sick people. Those are frightened people, influenced in other ways. Thank you very much.
Okay, thank you very much for the presentation. As you were presenting, we got concerned on whether you went ahead and gave them alcohol or you didn't give them alcohol, because we are looking at, were you recruiting participants who had taken alcohol? That was maybe the inclusion criteria. That's what we are thinking. But you answered it when you are saying that it was one of the limitations, that it was a dry run or there was no liquor. Now my concern is on the way forward. In research, we well know that we have to consider the ethical issues. You are going to carry out a study on this participant, but the participant is unaware, so you are like blinding him. So at the end of it, he might be skeptical and say it can be a legal issue again to us, and they say, please, who told you to take my information without my consent? I don't know how you're going to deal with that. Thank you.
Actually, let me come to the significance of this study. For example, here in Rwanda, we should be having some cases where people will just make consents, written consents, when they are drunk. Otherwise, they will try to disguise their handwriting, because they will say, like, I was drunk at that time, so don't consider my handwriting. So it is a very big concern, because we are having so many cases whereby when someone says, I was drunk at that time, and when you are taking the specimen, it will be a very big issue. So let's go to ethical issues in this research. One, I think we may be having volunteers as soon as we are not in a dry state, because when I was in India, it was a dry state. There was no beer, no liquors, because it is illegal to drink there. So I think here it is very easy to have volunteers who can sign a consent. I will be having, I will have my Uganda origin, like yours in Uganda. I will have 2 Uganda origin, then we calculate the blood alcohol content. Then we say, okay, you are good to give the samples. I think we may be having volunteers, and we will make sure that people will sign the consents and whatever, so that we can be safe during this research. Otherwise, there should be some other ways. But what I can emphasize on this is that the research itself is very important, because we are having so many cases. Because I'm a daily practitioner of question endorsement, even if these days I'm working in DNA, but we are having so many cases in such cases, so that we have to find out whether we can find some fixed criteria to be considered when you are taking the specimen and whatever. Thank you.
One question here.
Yeah, thank you very much. I'm by names of Chubero Karangwa Verita, medical student at University of Rwanda. And I wanted to ask about at what percentage did you find the handwritings likely to change? And if maybe you didn't consider that, it would be better in your future work to consider maybe introducing a neurologic physician to see to measure the neurology effects of alcohol, so as to know at what percentage it changes depending on the kind of person, because it's going to be different due to the tolerance you said. Yeah.
Thank you very much. I think I said it before, that the neuromuscular motricity will coordinate our handwriting. That's a principle. And then coming to your question, I think it is very important to work with, you said neurologists, right? I think it is very important to work with them just to make sure that our result will be based on factors that are very somehow scientific. So I think in the future, that should be a good idea. We have to work with physicians and neurologists just to help us to understand the influence of this muscular motricity that coordinate the handwritings. And then to your first question, it was there in the presentation, the overall change in handwriting was 60% changed their handwriting, while 40 maintained their handwriting. But I try to explain the numbers. One, we were in a dry state whereby before even recruiting participants was not easy, because you can't have a Hindu person and you try to give him alcohol. If you are saying, you see, I'm in a research, help me doing this and this, he will accept, but he will still be controlled by the emotions and whatever. So that's why most of them, they maintained. But I'm very sure that if we do this, like here in Rwanda, if we conduct the same research and we increase the numbers, I think there should be some deviations. What we'll be doing in the future may be different from the numbers we are having right now. Thank you very much.
One more.
Actually, as I said, the alcohol content, the blood alcohol content was maintained between 0.08 and 0.12. I think it was in the presentation. That was what we maintained for the people, and we had to calculate the blood alcohol content. Yeah, that's good. I think thank you for the intervention. I think in the future it's better to work with a neurologist so that we can have this idea of. Otherwise, what we're considering, the participants, some of the participants may be tolerant to alcohol, others may be drunk before, things like that. So people do behave differently when they are exposed to alcohol. So I think in the future we'll just develop our research together with the neurologist. We have also to work on the number, the size of the samples, things like that. Thank you very much.
And one neuroscientist here wants to comment.
Yeah.
Thank you very much for that wonderful work. And mine is like a comment in terms of the future work, because I'm talking at the level as a classroom teacher based on our experience with students when we teach. And again, I want to suggest, if you want to do the research here in Rwanda, or to develop a database on handwriting change due to tremor, or due to, we call it bench drinking, where people go to drink like 2 bottles of Skol or 3 bottles of Skol, but they are not really alcoholic. They just take that to relax. I want to, what we call the exclusion or the inclusion criteria. What will, like suggesting to develop future research with inclusion and exclusion criteria. You understand what I'm trying to say?
So you are trying to say like inclusion and exclusion.
Yeah, the criteria, like if you are going to do the research, this is a good research.
Yes.
In terms of, because we as teachers, we have students, or we have cases where a student can put, when you hold your pen like this, you have a different writing. When you put it in the second finger, you have a different, and you put it in the third finger, you have a different writing, because students use that in terms of writing tests for another student.
Yes.
You understand? So I'm just suggesting that in terms of the future study, there should be a inclusion criteria and exclusion criteria that student or that person or that volunteer must like have the pen on a particular region of the...
Yeah, I'm getting you. Okay. I think in the future we'll be working on that so that we set the same standard of writing. For example, you see when it comes, and again, you should understand that when we are considering the participant, should be writing in the same way as they have been doing it before. So we have their handwriting before, and when they are sober, and then you have their handwriting after drinking. So we will try to include all this in the future, and I think it is a good contribution. Thank you very much.
Thank you very much. I'd like to call Sazo. You can give him a round of applause. I think it was a tight one. Yeah. Sazo Murguiro, is he? Yes. So he's going to be talking about role and duty of crime scene investigator in the case of strangulation by differentiation to hanging. So. And I remind all speakers to adhere to 15 minutes. I will be cutting off now. Honeymoon is over.
Okay, thank you. My name is Sanduma Pere Celestin. I'm working at Rwanda Investigation Bureau as a crime scene response investigator. Right now, we are going to talk about role and duty of crime scene investigator to differentiate hanging to strangulation. This is the topic. What we are going to see, we are going to see the introduction, aim, differentiation, or characteristic that we rely on to differentiate hanging to strangulation. Case study, what you have really done is a retrospective study, and then we analyze, then we conclude. By the introduction, before, at the end of this presentation, is where we are going to see where we need specialists. A crime scene investigator has uncovered the physical evidence to help identifying what happened and who was involved. Investigators all the time is there to capture the suspect. This process must be conducted carefully, of course. We will take it carefully for never destroy the evidence. It is easy to diagnose hanging and ligature strangulation in the presence of characteristics that we are going to see. According to the ligature marking, the distinction between the two types is very important because strangulation is presumed to be homicide. It means if it is homicide, it is someone behind for those strangulation to make someone dead. Sorry? Okay. So the aim of this presentation is to determine the distinguishing feature of ligature mark in ligature strangulation, and it is to study the information provided by the ligature in ligature asphyxia death. Some characteristics that we are going to talk about are those, but the list are there, but not the list is not. On that, there are so many characteristics that you have to rely on when you have to differentiate strangulation to hanging. Some of them, there is a ligature mark, where the ligature is located, the form of the ligature that you have in the neck. For the hanging, ligature mark is oblique, means it forms an inverted V shape. And it is located at the higher of the neck, above the thyroid cartilage. In strangulation, the ligature mark is transversal, means it makes a whole circle in the neck, and it is located below the thyroid cartilage. Also, we have to see the abrasion, ecchymosis, or contusion, means there's a bleeding under the skin. In hanging, it is so less, while in strangulation, the ecchymosis or contusion, it is so more prominent. Sorry. The thyroid fracture, we have to palpate on the neck to see if the thyroid is fractured. So in the hanging, it is so common. The thyroid is there, is fractured, but in strangulation, it is not fractured. On characteristic of thyroid cartilage, it is less common in hanging and more common in strangulation because of where the ligature mark is situated. So on the tongue, in hanging, the tongue is more swelling and the protrusion is less marked, while in strangulation, swelling is more marked. Saliva, on the hanging, saliva is running out of the mouth, while in strangulation, it's so absent. Is what that, from those characteristics, is what a crime scene investigator must see out the skin, because a crime scene investigator only make external examination. So for the involuntary discharge, the feces can cool out in strangulation, but in hanging, it is not there. Here we are going to see some photos of those characteristics where it is located and even how it is look like. So as you see, for this strangulation, the ligature mark make a whole cycle in the neck, while in hanging, it make inverted V shape. For tongue, in hanging, it is not swallowed, it is less swallowed, while in strangulation, it is more swallowed and protrusion. In strangulation, saliva is absent, while there in hanging, saliva is there. The case study was a retrospective study that was on crime scene conducted from our team from the January to December. And in a duration of one year, we make 64 scenes for hanging and strangulation. And the study was to study for where age and the gender of the victim and the ligature mark with its reference site. In the 64 scenes attended, 51 cases were of hanging and the 13 were strangulation, with a group of age, as our study, between 20 to 60 years, with an incident rate of 78.6 and 20.31 proportional. It means 79 was on hanging and 20 was on strangulation. In that case, in 61 of hanging, 45 were men and 6 were women, with the difference equivalent related to 8.2 and 11.7 harmoniously. And in 30 cases of strangulation, 7 were men and 6 were women. The question is that why men are more hang themselves than women? So at the end of this presentation, someone will, from the psychiatrist, may answer for that question. Why men are so hanging themselves than women? So, for the conclusion, is not. The crime scene investigator is a crucial work to identify necessary evidence to help investigation. In the case of strangulation and hanging, scene investigator has to play a huge role to make sure that his or her findings must be helpful in prosecution. So in terms of strangulation, in the case of strangulation, a crime scene investigator must make sure he works with a specialist from the pathologist lab or from medical doctors. From those cases of strangulation and hanging, a crime scene investigator must consult a coroner, as I said, or a pathologist for a better clarification of the case. Due to those features, makes hanging to be suicide. Strangulation, hanging is suicide, means is the one who make, as I can say, who commit himself to be died. And strangulation is judicial, means it has to be reported to the prosecution. And it helps crime scene investigator to draft a report to be submitted to the judicial system for jurisdiction. So the draft of report that a crime scene investigator must draft, it can be made with those pathologists from those draft is also made by those pathologists or a specialist. So this is the thank you for your kind attention, question and comment. But my question is there. I would like to ask for psychologists why men are more hanging themselves than women. Is that anyone can answer for this question? Thank you.
Thank you. I don't think we have time for them to answer that question. Maybe it's another investigative question. I will take a question from audience 1 because you have your 5 minutes, 3 minutes left. So, question? No? Good, thank you. So, thank you very much. Let's give him a round of applause. We're going to switch a bit on the program. So we are going to hear from
dismemberment and dumping after homicide using setting in concrete. And you're welcome.
Thank you so much. Yeah, I hope you're still a little awake. It's warm and they skipped the coffee break, so I'm sorry. But maybe it's also an interesting case I would like to show you just now. If we have some pictures, and I hope you can see the pictures. Maybe this fits perfectly because we had heard so much about CSI crime scene investigation, and this is like a case where this crime scene, all the methods became really important. Thank you. So for all of us who don't know me, I'm Eileen. I'm the forensic anthropologist at the University Hospital in the Institute of Legal Medicine. You hear me? Okay. And I'm really happy to be back here finally after so many years here in Rwanda. And yeah, maybe you like my case I would like to show you. So, well, this is, can you see the pictures? What would happen? There was a 41-year-old man who was reported missing by his family. They said he just disappeared. They didn't hear anything about him. So he was just off. Sometimes this happens. Some people go away, get some cigarettes, and don't show up anymore. And then after 1.5 years later, a young man showed up at the police, and he stated to the police that his ex-girlfriend told him that her father got killed by shooting twice in the head and then buried inside an indoor riding arena. And here you can see this the property of the family, hopefully. Where's the pointer? I need my glasses. No, here. Here you can see the property, which is really huge. It's an area where not many people live around, so it's a little bit staying for itself alone. And here in this area here, where the arrow points on, you have the indoor riding arena. And this is the view from the inside of the arena, and the witness said that in this area here, the body was buried. So we had to remove, first of all those vans and all kinds of machines to have like a plain field of space. After this, after that, this is really too light, maybe you can't see it. I asked the policeman who joined us, like 50 policemen, young, strong men, to get rid of all the soil which was loose on top of the ground. So they used shovels, and then after a while, this rectangular structure, maybe you can see it here, appeared exactly at this area, where the witness pointed on. So then I started the excavation by using anthropological archaeological standards, and after a short while, it started smelling, and we found like a big piece of soft tissue in a way. But I couldn't really say what it was. And because the police found some cadavers of sheep on the ground on the property, I was not sure if this is maybe another sheep or maybe a human. And also, what you can see here, maybe, I don't know. Well, here, we have like a grave. We found like a grave, like 2 meters long and shoulder wide. So it was strange, it was a strange thing. And then we got this piece of soft tissue out of this grave. It was wrapped around with a rope, like you can see here maybe. And as we got it out, at this moment we could tell that it was a human torso. But also we could tell by looking at this grave, you can see it here, that the whole body was inside this grave first, covered up with slaked lime or limestone. And by looking at the imprints, you could tell inside this concrete block could be just garbage, rubbish, because they did some construction work inside those buildings, the house. But then they found another one, and at the end we had 6 of those blocks. So then the question was maybe the missing body parts could be inside those blocks. But how to get those blocks out, and we asked ourselves, how could we look inside? So I called my former head, Mr. Pische, and asked him if we could just bring the blocks into the institute and do some CT scans. But then, because it was a little bit too complicated, we asked our colleagues from the German custom departments if they could bring their mobile device. It's like an X-ray device, it's called scan van. Usually they put in some suitcases to see if there's some drugs inside or maybe weapons. And brought this to the scene, and we put one concrete block inside, didn't really know if you could see anything at all, and then this is the one block, the first block, and here you can see already, this is a lower leg and 1 foot. So it worked perfectly. So at the end we had, we found, in all those 6 blocks, the body parts. We could detect them first with a scan van. Then we asked ourselves how to get it out. Because those blocks were so heavy, we decided to open them up at the scene, at the site, with heavy machines, and I can tell you, those police officers, they really love to do this with those heavy machines to open them up. But then as we got closer to the soft tissue, we had to use a hammer and a chisel to prevent the disturbance of the soft tissue, especially as it got to this one block where the head was inside. You might see it here. So at the end, this picture shows you we had those 6 blocks, and in each block were some piece, 1 or 2 body parts of this man. Of course, we brought all the pieces to the autopsy, and then the legal medical doctor did the autopsy, and first of all we could show that these are the remaining body parts from the person, the man who was previously missing, and we could also not just identify him via looking at his tattoos he had right here on the arms and there, but also via DNA analysis, of course. And also, they could confirm the cause of death. And here you can again see on the left-hand side, this was the image from the scan van on site, and here you can recognize 2 holes or something, 2 dots. And after we did the CT scan and of course the autopsy, we could confirm that this man was killed was shot in the head 2 times, one from the back and the other one got through the right parietal. So this was really quick now. Maybe it was good not to talk too long to you. I wanted to show you with this little case how important it is to do a proper excavation on site and to get all the information you need later on in front of the court when you have to explain what happened, because this way we could say, all right, first of all, the person was killed, buried inside the riding hall, got out again after an unknown period of time, cut into pieces, and was dumped in concrete in this little creek. And also, it's really good to be a little creative maybe sometimes when you're at the scene or at the site, and maybe you have to think about new ideas how to get more information about what happened. Not everybody has a scan van, and not even in Germany, but sometimes it helps if you can use maybe creative ideas because of the infrastructure, for example. So that was it, real quick. So if you have any questions. Well, I have my anthropological workshop tomorrow and the day after, so maybe if you like, you can join this workshop. That would be really nice, and then we can talk about one and the other topic.
Thank you very much. Please give her very, very warm greetings. Quickly, we are going to welcome the next presenters. The next one is with Lekata and Fabrice Radukunda on study, conservation, and exhibition of human remains, the need of bioethical perspective
I don't know, Fabrice, if you're here. No, okay. So then let's go to the other presentation on Murambi Genocide Memorial, preservation of restored bodies as a final report with Martin and the team.
Thank you very much. My name is Martin Moza. I'm working with the new ministry, the Ministry of National Unity and Civic Engagement, Minubumwe. Before joining that ministry, I was working with the National Commission for the Fight Against Genocide, former CNLG. It's a pleasure for me to give this presentation with my colleague from Germany. Here we have Monica and daughter. They came from Lower Saxon State Service for Cultural Heritage in Germany. In this presentation, of course, it's a big project we started since 2016. Even before that period, we worked as the National Commission for the Fight Against Genocide, working in the preservation of human remains across the country. So during that period, of course, we started a project, and since 2016, we came across to meet exactly Professor Pusho via Professor Mutesa from University of Rwanda. Then we discussed the project, and finally we continue together in that project. The project is about cleaning, conservation, preservation of human remains at Murambi. As you can see on this picture, this is a picture of Murambi. Murambi is a genocide memorial site among different memorials across the country. As I said before, I was working with the National Commission for the Fight Against Genocide, and now that commission was replaced by the new ministry, Minubumwe, the Minister of National Unity and Civic Engagement. And in that ministry, of course, we continue with the mission accomplished by the National Commission for the Fight Against Genocide, which are the maintaining and preservation of human bodies and which are found in different memorials in the country. But for this project, we focused mainly to Murambi Genocide Memorial site. Why do we need to conserve human remains and other genocide physical evidence? We preserve purposely to learn from the past, for the present, and even for the future. So those physical evidences, even human remains, which are found in the memorials, help us exactly to learn from those physical evidences. Next. Oh my gosh. As you see on this picture shows exactly different memorials. We have more than 200 memorials across the country, but here we can see different memorials. This is the Kigali Genocide Memorial Site, Bisesero, Nyanje, Murambi, which we are talking about, and Ntarama, Nyarubuye, this is Ntarama, this is Nyamata, and also different memorials across the country. Among those 200 genocide memorials, we have 8 at national level, which are under the responsibilities of the Ministry of national unity and civic engagement. As you can see on this picture, we try to show exactly different physical evidences we can find at Murambi. Here you see exactly the human remains, they are the bones, and here we have different rooms which are accommodating more than 800 human remains preserved by using limestone. And here it's a main building exactly of Murambi. And here we have some textile, we have some cloths, and also those cloths are mixed with different materials we found exactly after genocide at Murambi Genocide Memorial Site. How the idea of preserving the human bodies came out. As I said, when we started the National Commission for Defense Against Genocide, there was an idea of preserving the rhymed bodies found at Murambi. By having that idea, we were not able to put into practice the project until when we met Professor Pusho and his team from Institute for Legal Medicine in Germany. Then we started working together. From that period, we tried to see among the buildings which were available on the memorial. This was a big hall supposed to be the dining room, because Murambi was a technical school under construction during the genocide. So among its buildings, we found a big building, a big hall, which was supposed to be the dining room. So we said it's better to renovate it and then to rehabilitate it, and then we can put all those the dead bodies rhymed into that hall. Of course, with the Professor Fusho and his team coming from Hanover, we tried to develop quickly a project doing some tests and see how we can proceed exactly to preserve those human remains. We selected 20 bodies, and among those 20 bodies, there were also young children, adults, women and female, exactly in order to see how we can make a good preservation and also a good representative presentation of those 20 bodies selected. This was, as I said, this was the dining room for the students. And then after rehabilitation, and this is how the room looks today, with the different, of course, presentation of human remains inside. So I'm gonna pass the floor to my colleague, Monica, to continue exactly explaining the achievement during this project, during the time we spent at Murambi, working on 20 bodies preserved at Murambi. Thank you.
Thank you, Martin. Just to let me quick know, next slide is this, and this is, okay. Okay, okay. So again, thank you very much, Martin. As you can imagine, until the conservation hall in Murambi looked like this, it was a long, long journey. So let's take a quick look back how our involvement came about and why we as archaeological restorators are involved in this project. So in archaeological restoration, we are always dealing with a variety of materials and objects. On this picture, you can see some examples. In addition to inorganic finds such as glass beads, or gold jewelry, there are a variety of organic finds made of leather and textiles as well. The preservation, including to make the history and information which the objects contain readable and accessible, is always the central purpose. A lot of these objects, especially those from ages without written tradition, like Bronze Age or Iron Age, came from burial context. Therefore, it's no wonder that in our work, we have often to deal with human remains themselves. These can be skeleton finds, as you see, or cremation burials, but also entire bodies such as mummies or bog corpses, which are still preserved with skin and hair. Oops. Like the bog mummy, Red France. Through the work with bog bodies from Lower Saxony, our institute can look back on a long and intensive cooperation with the Institute of Legal Medicine. So thank you for our cooperation all the time. So it's the colleagues from Hamburg to whom we owe our participation in this international project. During their stay in Rwanda, they were confronted by CNLG employees with the question, how can we clean human remains treated for a long period with limestone powder? When you see this picture with the two images, it's more than understandable why. On the left, the girl of Ictr, a 2,000-year-old bog corpse, and on the right, a victim of the genocide 20 years ago from Murambi. Although the remains from Murambi are neither from an archaeological context, nor are they completely artificially or naturally preserved mummies, the methods for treating human remains are still similar, and they are always based on great respect. Our introduction to this project started in June 2016, when, after returning from Rwanda, a forensic scientist from Hamburg, Eileen, called me, Monica, and asked, how do you know how to clean and preserve human remains that have been treated for a long time with limestone? Of course, this question sounded simple at first, but I tell you, it isn't. We were supposed to develop a concept for cleaning the human remains, a concept for their long-term conservation, and as well a dignified presentation, but also the transfer of knowledge to let you know what we do and why we are doing it was very important. And there was only a really short time frame because there, in April 2019 was a big 25th anniversary of the massacre in Murambi, and a big commemoration day, and our will was to be ready by then. And last not least, as Martin shortly mentioned, we had to use existing resources, specifically the acrylic coffins you can see on this picture, and as well technical equipment purchased in the previous project. Although in our profession we regularly deal with human remains and have to clean and preserve them, this whole project was a fairly new task for us. No two artifacts are ever the same. Methods that applied to one may be completely wrong for another. So when we talk about the cleaning and presentation of the bodies from Rwanda, it means that first we had to find the right method for removing the limestone powder. And in the end, we come to a good solution by using airbrush with cork powder. But on site, we were confronted with another phenomenon, a layer of fat wax. This was possible to remove with dental tools. For successful work on site, it was essential to define the needed working process, like pre-cleaning, weight control, photographic documentation, written documentation, then cleaning again, an everlasting circle, always ending with regular monitoring. But more details about the working station and our work on site will, you can now hear by my colleague daughter.
Thank you, Monica. Yeah, well, these keywords you see here represent the different working stations we developed over time. It's the final setup, because we have been to Murambi 5 times by now, and just like the cleaning method, we had to, refine and work on it to be most effective. How this looked in real life, you can see here. Here, the pre-cleaning took place. We used a vacuum cleaner and soft brushes just to remove the loose deposits and to facilitate the handling of the bodies for the further processing. The weight control using a digital scale with an exactivity to up to 1 gram is essential, because it can give us an indication about the condition of the body. If the moisture content changes, it will certainly show in the weight. So the weight control as part of the documentation is essential to evaluate the condition of the bodies. The documentation with photos and also written words is always an essential part for us conservators, to just have an assessment about the treatment, but also about the body before and after and during the treatment. So before we started the main cleaning process with the sandblasting machine, like Monica described already, we had to document all the bodies. In Murambi it was even more important because it also included the forensic analysis, which we were greatly supported by our colleagues from the UKE. So after the first documentation, the main cleaning took part. It was a very demanding process, but I will come back to that soon. And after the body was cleaned, we documented it again, because only after the cleaning of the limestone powder, most details were certainly accessible or even seeable. And as Monica said before, for the long-term preservation, the monitoring on a regular basis is essential. We decided during the development of the conservation concept not to use any chemicals or conservation agents. So regular monitoring, looking and documenting how the bodies react now that they don't have this protective layer of limestone is essential for the long-term preservation. So now, because the process of cleaning the bodies with the sandblasting machine was quite demanding, I will show you soon a small video. But first, the process of cleaning was carried out in a specially built cabin. We had to use protective equipment because of the high dust development, and also it was quite a loud process with a lot of noise pollution, because we had the sandblasting machine coupled with an air compressor as well as a vacuum cleaner. You can imagine that's quite a lot of noise, and if you sit in this cabin wearing protective gear, it's very demanding. So I hope this works. Nope. The movie? Yeah. So just to give you a short impression how that actually looked on site. And now it's not working. Nope. That's too far, quite too far. maybe we can just skip the movie. Yeah. Nope. No, I don't think it's working. But that's okay. We just skip the movie, then we are earlier finished, and everybody will be thankful for that maybe. No. Okay, I just take the next slide and continue. Okay, imagine you saw a movie how the process of cleaning actually worked on site. I think this slide shows exactly why the cleaning process was so essential also for the forensic analysis. There were a lot of details which could only be seen after the cleaning, such as here, fingernails and even the textile impressions, the fabric impressions on the skin of the victims was visible after the cleaning process. And also, sometimes even the gender and age was was easier to determine after the cleaning process. So I said the cleaning process was vastly supported, not the cleaning process, but the forensic analysis was vastly supported by our colleagues from UKE. They distinguished between the ante, peri, and post-mortem injuries, but they also catalogued or distinguished between blunt force trauma and sharp force trauma, and of course they catalogued injuries with a certain intent, such as cutting the Achilles heel so the victim couldn't run away, or defensive wounds like cuts on the underarms where the victim tried to defend him or herself. So all these processes, the cleaning, but also the forensic analysis, and as well the presentation concept served the purpose to, show the individual history of the victim in more detail, to give them a voice of what happened in Murambi so many years ago. So, as Monica said, we had a short time frame to prepare for the actual exhibition for the 21st April 2019, and we had a lot of handiwork to do as well. Here a few pictures from the preparation work. And this was then the big commemoration ceremony where a lot of people came to remember the victims and also see the new setup of the individuals shown in the conservation hall. So this was the review of the project. Now I give the word back to Martin, and he will tell you what happened since 2019 and give a quick outlook how it will go in the future.
Thank you, Doctor. Since 2019, a lot of things happened exactly at Murambi by innovating the memorial. As you can see, this is the main building. We tried exactly to rehabilitate the compound and trying to write the names of the victims near the mass graves, because we found more than 5,000 names. Then we continue to extend exactly the list as we continue to find the names. And here we try to put also the names of the perpetrators, the big fish, exactly like the prefect Chibaruta, who is now in trial in France, and others organizers of the genocide at Murambi. And also we try to put the signs on the different buildings showing exactly what is inside of those different buildings. We try to maintain exactly the exhibition hall, which is the conservation hall, by putting the curtains in order to avoid the right sun to enter inside for destroying exactly the human remains we preserved inside, and also protecting the acrylic coffins we used in that conservation hall. So for the future, we have exactly to continue maintaining the preservation hall by maintaining exactly the acrylic coffins, by continue making monitoring, inspection, documenting, and also by noting any changes on the dead bodies we preserved in that hall, and also by continue extending the project and the cooperation between Rwanda and the Germany team. Before concluding our presentation, dear participants, allow me to ask you to join me to give a big clap to Professor Pusho and his team, because they did a big work at Murambi, because since 2009 and even 2008, we tried to see how we can preserve the human bodies at Murambi. We tried many times and we failed, but once we met them, exactly we found a solution for this challenging historical facts in Rwanda. So thank you for your kind attention. This is our photograph we took yesterday while working from Murambi. Thank you very much.
So a quick question. A quick appreciation of all a round of applause to them. Because they did, I think ever since, they have done a very, you want to say something? No. Yeah, so I was saying, I wanted to make a special, because I think they have done a very wonderful work. So let's give a hand clap that is also with some rain. Do you know rain? Yes, so you're going to clap in the way of rain. Quickly, and then it stops. You know how the rain goes? It starts slowly, increases, and then it goes slow, right? To the team, right? So let's go. Thank you very much. The next two presenters, one, they sent in, they are not here, but they did their virtual presentation. So we're going to have their presentation. One is Adelite Mukamana.
Good afternoon, everyone. I'm happy to be with you today. My name is Mukamana Adelite. I'm a clinical psychologist and an narrative practitioner. I work with individuals and the groups, and I'm in charge of clinical supervision in different organizations. Today, I have a pleasure to present to you the work I have done when I was working with children born out of rape during genocide against Tutsi. My work goes on ways of living and survival by children born out of rape during genocide against Tutsi. Now I'm sharing with you my slides. Yeah, as I told you, I worked on ways of living and survival by children born out of rape during genocide against Tutsi. And as I was started, while working with MICA, the organization that support and advocate for survival of genocide in Rwanda, I met many children born out of rape. And during the group session with these children, men talked about their relationship with their mother, their relationship with the rest of the family, and the relationship with the rest of the community. One of the things that was difficult for them was to live in a community where they are constantly considered as a reminder of traumatizing experiences of their mother. The objective was, first of all, to learn hardship moments that children born out of rape went through after genocide, and learn ways that they used to manage those hardship moments. Setting. Then I'm going to present to you the results from my interview. And the first thing I'm going to present to you is that, first of all, the hardship moments faced by the children born out of rape. Those children faced, first of all, what they call identity problem. As they claim not to be considered as a victim of genocide against Tutsi, and therefore they don't benefit from anything that survivor deserves, such as school fees, while their mothers are so poor that they cannot afford themselves school fees, or other social basic for their children. And those young men and women and the ladies was talking about scapegoats being scapegoated by the community for all problems that their mother facing. One says, my mother don't love me because she considered me as an obstacle to her life. She told me that I was the one who made her life complicated. And for her, if she didn't have me, it would have been easy for her to access studies and to be like others. Another male participant said, other children are called children of blessing, while we were called children of curse, like memories of the grief and the sorrow of our mothers. Another problem presented by the interviewees was facing stereotypes and labeling. They are not loved. They say that they are not loved in their mother's family because they are treated as children of killers in their family. And those children are split into two categories or two groups. Some of them are called children of Ictr, lose the meaning attack, because their mother were raped by so many people that their children cannot know their father, because even their mother do not know who raped them. And others called the children of Ababo, because their mother was raped by one, it's just a single rape, and their mother know where they are raped and who raped them. And the mother were raped by the person who had took them hostage, and they ended up getting pregnant. Another challenge is that sometimes they are considered as a source of conflict. Some of those children have come to know their father and even know that there are some who are in prison because of the witness, the testimony of their mothers. And sometimes those children want to know their father, including visiting them in a prison. And that causes conflict between the children and their mother. And they express also being a victim of their background as children or as child born out of rape. One of the female participants in the interview testifies that there was a boy who made me fall in love and made me pregnant. And I felt it would be okay, but when we were preparing to get married, he learned that I'm not a survivor in a sense they considered a survivor. Instead, I was born out of rape. He immediately declined and told me that he could not live with Interdisciplinary, and now I'm raising alone a child. But at the same time, if those children, they're born out of rape, now they are young ladies and men, facing different hardship moments, but they tried also to develop some hope, some resilience, some coping strategies that help them to go through these difficult moments. And using the developed framework for receiving and documenting testimony of trauma by David, I asked the following questions that helped me to understand how they got through this hardship moment and how they managed to face all these problems in the community where they live. And my first question was, what does this say about what is important to you, about what you care about, and the value in their life? As I taught you, to keep the secret of the respondent, I used the acronym. And the key A in this, what I call key A in this interview said, I feel the most important thing in my life is not to give up. UD said, I think the most important thing in my life is to know that your history should not define your life. It is up to us to choose the right life, even if the problems are inevitable. And LE says, the thing I value in the life is to make sure that what happened during the genocide don't happen again. I will give my life if necessary. BG says, I feel like I have to strive for a better life and not to be a hostage of my life. It's a real powerful expression, not being hostage of my story. To continue my interview to better understand how they cope with the hardship moments, I asked questions to know things that was been always important to them and what's the history of this. UD said, I grew up and found out that my mother had a conflict with her family member because she refused to have an abortion after she was left during the genocide. When I grew, I began to feel that I had to overcome my history, succeed my studies, and strive for better life. And UG said, I grew up and found myself living with my mother alone. I come to know that she was raped, but she changed my birth certificate so that I would not know my history because she told me that my father died when I was a kid. Unfortunately, I ended up knowing my history. Later, she told me that she did it to protect me. At first, I felt that story unbearable, but later on, I thought that even though I was not informed before, for now, I feel like it's no longer harm me, even though my mother is always in trouble with her trauma. During the time when asking the questions, I was thinking about asking when those children began being subject of this injustice, how did they try to endure this? What did they try to think about? Where are the memories that they tried to hold on to and dream? What sustained there through those most powerful times? And here he responds that after I found that it seems difficult to live with my uncle, that is A.E. And she added, I got a chance to succeed my exam and went to high school and stay there during my holidays. I had a dream that when I graduate, I'd go and help the children who need, and I would help my mother get out of her home and find a place to live in peace. I graduated. The first thing I did is looking for a place where my mother can live. And now there are also some children that are happy with small things like getting books and buying for them insurance. This testimony demonstrates that being helpful for others may help someone to go through the hardship moment, to feel important and useful for the society. DG says, I got a chance that my mother cared for me, even though she tried to hide my history, but she loved me so much. I'm the only child she was, and she gives me the strength to feel that I am the one to take care of her. Her mother has depression, and she's very close to her, and she thinks she will get a job and help her mother. Another question was about different ways that they tried to endure the different forms of the trauma. And LE says, one of the things that helped me was to go to school and find a place to live. Another thing that helped me was that my mother loved me, even though she didn't have much ability to get me out of the problem I was in. When I was in school, I went to the ERG family, which made me feel like I had another family of people who cared about me. The Compassion organization also helped me pay for school fees so that I could study without focusing on my son. BG say, the first person who helped me is my mother because she always strives for a better future. She has not secret from me since she has confessed to me about her rape during the genocide. Even though she didn't want people to know about it because she thinks that they would stigmatize me, she empowers me a lot and capable and confident that I will achieve more. And another question in my interview was, why is it important to them for other people to know about their history? You say it is good that people are aware of the history of our parents and the impacts it has had on others, so that even those who were afraid may be able to dare to make their testimony public. Even now, if our parents did not give evidence, we would not have received the help to go to school. Avega, it's a widow association for female survivors of genocide against Tutsi in 1994, has also been very helpful to our parents in treating trauma. So I can feel that if they don't know their problem, they wouldn't be helped. GB says, testimony is good because it is one of the proof that the genocide against Tutsi took place. So that help us to know how to fight and better those who deny it. I asked them that at the beginning of the interview, they spoke about the things that are important to them in their life. And my question was, how have you been able to keep in touch with those values, those hopes for the world, despite the abuses that you were subjected to? You said, I like to listen to others a lot. Every person, even if I don't give something, but I listen to him, and that is comforting. LE said, I like to help others because I am where I am, because they are those who help me. TA said, I feel happy to help my mother because she also cares for me. When I will get children, I will not hide anything from them because I had found it difficult not to know my history. BG said, what I have done so is that there are things that no one can change. What is important is to know how to deal with them in a good way. I was asking if they have been able to reduce the effect of trauma in their life and how. You say, one of the things that helped me in their life, it makes friends. And LE said, when I got to high school, I went to ARG family and I had a friend to talk to. Those who would help you when you have a problem. I had another opportunity to compassion organizers face calls for me, and I felt much supported. I feel useful for the people because there is some who I helped, and I like to talk to other young people in a cooperative. And KA says something that gives her strength is to help her mother. She said, there when she has trauma episode or other illness, I'm the one who has to help her in the household and have physical and emotional strength to survive. Another one, BG said, the first thing that gave me strength was my mother's love. And I was asking if there is ways of reducing the effect of trauma that they have developed newly, and what's the history behind the new way of dealing with or reducing the effect of trauma. You say the new ways of reducing the effect of trauma for her include going into activities that connect to others. She said she's a representative of gender at her college, and she often trained herself to help others. That boosts her confidence and the confidence of others on her. LE says, the way I tried is to share the knowledge I have with others. When I meet with other young people in a serving cooperative and one group, I help others to think about a project that can help us to improve our livelihood. KA said, a new way that helped me is the church choir. I spend time with others, playing, singing, and I feel happy, forget about things that hurt me, and I feel that God is on my side. EU said, I'm the president of the Businessman Association in our community. People trust me, and the fact that they choose me to be a leader as a woman is a testimony to that they believe in my ability. And I was asking if there is a particular person who have made difference in their lives. And UD says, the transformation of my mother after the help she got from Africa and Sebota has allowed me to have a relationship with her even now. Even when I am in trouble, she supports me a lot. I'm still in school and she's the one who is raising my child. That is the main reason for the change in my life. He said, the important person who changed my life for the first time is my mother. She helped me to find someone to pay a school fees. And he had started, and also he found IOG community, and he felt supported. And he has people that accept him warmly, and they help him to endure. If we consider the stories behind the hardship moments of those children born out of rape, we may see things. Those children, they have been supported by the community, they have been supported by their mother, or they found support in churches, but also they feel comfortable helping others. Their stories were full of skills that may help others to understand that even though those children faced hardship moments and many challenges, but they found ways to overcome and they continue to live better. Their testimonies for me can bring also changes for those who especially face the same problem. Thank you very much for your kind attention, and have a good day.
So this is for Samuel Habimana. He's also doing his PhD, and he's our last presenter.
Good morning, everybody. This is my great opportunity to be here again in this conference virtually. It's my great opportunity to share with you and disseminate my research that I conduct here in Rwanda. My name is Samuel Habimana. I am PhD student in the United States, in California, Rwanda University. I conduct a research with my colleague about the green space in mental health Kigali City. So I can share you my presentation so that I can go ahead with. As I said, the study is a potential of green space to address community trauma and mental well-being. It's a study post-genocide circumstantial in Kigali City. This is Rwanda, you know, this is Rwanda, this Kigali City. This is the authors who are part of this research. I'm Samuel Habimana with those friends, colleagues. To introduce in this presentation, I will talk about the introduction, literature review, gap, objective, methodology, ethic issue, result, discussion and limitation, and conclusion. Introduction. Mental health is a wide-world public health problem. 1/4 individual has had mental health disorders. More than 80% of the world population lives in low middle-income countries, where Rwanda is included. The mental health may be adversely affected by a history of civil conflict, poverty, violence often found in lower middle-income countries. To address the prevalence of mental health disorders, include substance abuse, issue of trauma, investigators have increased a focus of potential of non-based solution, which is green space, especially in urban green spaces. According to some authors, like Brian has said, the green space contribute in the recovery of mental fatigue, but also the Modigi said green space is also contributing in mental health and well-being. The authors was showing how the urban green spaces have psychological healing, recreation, and positive behaviors. The method of the depression and physical stress indicators. These are different. Our research has been conducted with the impact of urban green spaces on mental health. In Rwanda post-genocide, there was continuation of the mental health problems as the effect of the genocide aftermath. Much research has been conducted in Rwanda to address this long-term impact of the following the genocide. High prevalence of trauma and transgenerational trauma has been documented with the research of Dr. Mutesa, Professor Mutesa and his colleague, and Celestine and his colleague. They highlight how the potential transgeneration of trauma in the Rwandan population. Community intervention has been implemented to address these trauma. That is a different community interventions like community resilience model. There is a laughter yoga, community-based social therapy. There's a different intervention that has been conducted in Rwanda in post-genocide to address this problem of the depression and trauma. The effect of Rwandan government, the Rwandan government has been done in many different activities so that they can help the Rwandan to overcome with the mental well-being and enhance the sustainability of development. There is intensive urbanization of Kigali according to World Bank, yet also the leader green bloc. Rwanda, 100 hills in Rwanda has banned plastic bags for ecological restoration. And Kigali planting the trees. This is also a great policy and project for trees and flowers to enhance the city livability, and environment resilience. Strong emphasis in healing and reconciliation. As well, in post-genocide, Rwanda has increased in the healing and reconciliation. But the big gap is how much have been, despite of those done in environmental advocacy, little attention to be paid in a lot of urban green spaces. Thus, this research has come up to show the impact of urban green spaces like parks, like forests, like gardens that we have in Kigali City, how those can have potential in improving the mental well-being of Kigali City community and as well as whole country. This is the Garden of Memory. I'm sorry, my picture is for Eri, but hopefully now this Garden of Memory, I think it's not like this. It has been grown. The garden has been growing. It's also a great initiative for an impact of urban green spaces. And also, it's near here behind near Nobreza, small miles from here to this Garden of Memory. My objective for this study, we explored how the Rwandan community, specifically in Kigali City, can access urban green spaces, reason for limiting use of urban green spaces, and relationship between urban green spaces and mental well-being, the methodology. It is a cross-sectional study quantitative approach. Our 422 participants were interviewed to participate in this research. We have been setting some inclusion criteria like presence in selected green spaces, being able or being 18 years old, ability to communicate and consent to participate, and approval was the approval for conducting this research has been first approved by University of Rwanda, IRB, College of Medicine and Health Sciences. Also, this research has been approved by Rwanda National Ethics Committee, Arendt, and also as well as Kigali City has been approved to conduct research. Yeah, as you see, we have been selecting 3 green spaces. This is the Fazenda green spaces. Many people visited these Fazenda green spaces. They go for relaxing, drinking, jogging. You see, it is a top mountain of Kigali. There is another green spaces called the Nyandungu green spaces. These are big green spaces where when we are collecting the data, we took this swamp of Nyandungu, eco space, we take the Restoration Church garden, that is place of Nyandungu. We were motivate to know the impact of those people who visit these green spaces. So also we use golf green spaces. Also you see some people go inside in a field of golf and go around by doing jogging, walking. So we would like to we interview them to know the impact of these spaces and their mental health. The instrument we have using social demographic questionnaires containing psychological, physical, and other patterns. We use also mental well-being questionnaire, which we're accessing the mental of the Rwandan population, specifically those who visit these green spaces. Study analysis, the descriptive statistics with demographic and reason for non-use of green spaces were done. We evaluated linear regression analysis to explore the association of the dimension of green spaces and as well as green space. As well, we also use a multi-logistic regression analysis by using odd ratios to see the impact of those who visiting the green spaces and the mental well-being. The results in our participants, 57.3 were male, 53.8 were younger than 40 years age, 37 and 22 respondents were private and self-employees. The mental well-being, which is very interesting here, those who visit green spaces who have been highlighting in Kigali, 91.5% they have a positive mental well-being. Another 8.5 have negative mental well-being. Also, the accessing the 82.2 receive they visit the green spaces because of they want to do jogging, social events like anniversaries, wedding anniversaries, going to play like a massage. It's a green space which is in a restoration church. Many people visit that green spaces for the praying purpose, and the girls and boys for marriage proposals. 39.6 respond that they are owning their own gardening and the green spaces at home. 34.1 live too far from their home. 25.1 bad access to the green spaces. 18% say the green spaces is dark and they fear to visit. Those are some reasons for not accessing to green spaces. 34.1 they say, oh, we can't go to green spaces because it is too far for us. 25.1 say, oh, bad accessibility. 28.2 say, very space while too crowded. Yeah, so those are different reasons that are limiting the population from Kigali City to accessing green spaces. The reason for accessing the green spaces, based on we have seen that concerning to Nyandungu, the 41.2 responded that expect to explore the quiet place. 42.2 they say they go in green space in Nyandungu for exploring the quiet place. Also on the golf, 43.6 say they respond that these green spaces to enjoy flowers, plant, and visit a quiet place and a peaceful one. Regarding to the third green space Fazenda, 33.6 said they responded well wanted to enjoy flowers, plants in Fazenda. Logistic regulation unit, we have seen that enjoying green space, landscape, having is significantly associated with the mental well-being with odd ratio to 2.9. Also, those who have been view the flowers in the green space, also they have been significantly associated with the mental well-being with the 3.7 odd ratios. And also those who enjoy the team spirit when they're doing the green spaces, sports in green spaces, walking, jogging, they are 2.4 times for mental well-being compared to those who don't do that in green spaces. And also, some do photograph and artistic work in the green space. Also, it's significantly associated with well-being. Discussion. Study indicated that accessing green space in Kigali city is correlated with the mental well-being. With other different research has been conducted in different spaces, different places. Those research are in the same line with our research. We showed how the green spaces are really contributing effectively treatment for the stress, depression, and PTSD. Also, there is the research indicated that over 91.5 participants have mental well-being, which is very interesting. These results we get from those who visit the green spaces. Their mental well-being is more than the mental well-being of the people who are from Kigali city, according to the research conducted in 2018 with the RBC, Rwanda Biomedical Center. Also, this mental well-being for the people who are accessing green spaces in Kigali, 91.5% is a high prevalence to Rwandan population. Is good. It has a great score of mental well-being over the all Rwandan community. What does it mean? This means that at least those who visit the green spaces contribute to them much, so that their mental well-being be well compared to others. I compare those others based on the previous research has been conducted in Rwanda, like specifically in Rwanda, which shows how the depression is 12, PTSD, trauma in total population 12, and genocide survivor 35. That shows that the high mental health problems. But there's those who visit those green spaces, their mental well-being is very nice, which we thought, according to the data, probably accessing to the green space can contributing to their mental well-being. Also, some identify the barriers to reach to visit some green spaces in Kigali by saying like in Nyandungu, Fazenda, or those green spaces. Some say the lack of time. Some green spaces they ask the entrance fees. Like Fazenda green spaces, you need to pay at least 3,100 francs or $3 to enter. Yeah. So those are some barriers for the people to access those green spaces. Attractiveness of the features such as a house broker riding flower may be exposed more people to the physical and psychological benefit of visiting the green spaces. The presence of gardening, flower sports, and social activities in green space also are associated with mental well-being. Our study have reported the similar findings. Those reported that exposure to green spaces, garden, open spaces, enhance mental well-being. Discussion to maximize the benefits of investors. Natural-based solution happening in green spaces are related to community, particularly mental health. It is useful to be broader approach of urban green space to generate population, make more inclusive and accessible, and important to urban planning in the rate of planning the urbanization. Due to the high prevalence of mental health problems in post-genocide, community-level approach to enhance mental well-being does not require a therapeutic commitment to scale up many people, and non-stigmatizing interventions such as visiting the park, green space can be prioritized in Rwanda. The strength of this study, current study is innovative, yes, and this has not been studied in the country of Rwanda where they study the impact of space on mental health. The results suggest that even the people who visit those spaces have better mental well-being than the general population of Rwanda, which is their well-being was associated with various aspects, which is including the benefits in spaces. The potential for positive impact of the affected person should be studied. The limitation, the data collection was collected in COVID period, which is limiting or causing many people to have stress. Urban green spaces were not as well as accessed as during the normal time because it was in COVID. Sometimes they open, another time they close because of COVID. That was also our limitation. In addition, a cross-sectional study design as well, it was enabling to explore the causality between the green spaces and the mental well-being. That's why we are recommending longitudinal research is definitely needed. To conclude, high education and high income was associated with better mental well-being. You also find that relation between various types of using green space and mental well-being in the rest of the further research. We are suggesting to do the green spaces an important piece and quiet press, jogging, and other experiences. Some barriers accessing the green spaces should be addressed, including lack of time. Urban green spaces should be expanded in Kigali and made available to the residents for free, lower cost as possible, so that the people can benefit for this impact of green spaces. So I thank you so much, and I welcome some questions and comments. Thank you.
Thank you so much. I know Samuel is following. Okay, so that brings us to our long day. I would like ask that we go through the last session, which is the workshops for tomorrow. And I can quickly announce them as they follow, then we can go home. So for your information, we're not starting at 9 as we did today, tomorrow. We are starting at 8. And all people, you don't have to come here if you're going to Kachiru for autopsy or for toxicology or for blood pattern analysis. You have to go straight to Kachiru. People who come here are only those who are coming for PTSD, forensic anthropology, and cornea transplantation. Does that make sense now? Okay. Then, for some workshops, as I told you, we divided you in 2 groups. So people who go in autopsy in the morning, if I read your name in autopsy, in the afternoon you're not going to autopsy again because it's the same thing they are going to do for the other group as well. So you will come, go for forensic anthropology here. So when you come for lunch, you're not going back to Kachiru, but you're going for forensic anthropology in the afternoon. And then those who will be in forensic anthropology in the morning, after lunch, the buses will take you to do autopsy practical session at Kachiru. Then, for blood pattern analysis, you also be at Kachiru, so you come, take lunch, and go back. Cornea transplantation, you also be here for the day tomorrow. You will go for autopsy then the following day, because cornea transplantation is only one day, which is 15th. We won't do it on Thursday. PTSD, you will be here throughout for the 2 days, tomorrow and Thursday. Yeah, and forensic anthropology will be in the hall. Let me get the name, but it's just down below. So when you are here, you can ask where there is forensic anthropology and we direct you. Okay, so I'm going to read out names. Please take a note of where you appear, and we want you that you go where we have allocated you. Because if you go there when you're not in the list, they are not going to allow you to go in. Just because we have planned this with... So for group 1 in autopsy, Eric Saramba, Viziaremye Patrick, Mutamba Masita Sarah, Kamanda Ange Pascale, Kananga Wilson, Hezidgayo Damasen, Kalemanjingo Noel Olivier, Donata Wineziyaremye yego, Isimbi Natasha, Shumbusho Ezekiel, Mbugwize, Uwase Ange Noela, Chuhailo Kevarit, Alistide Migabo, Nina Carol Rangira, Sesan Godfrey, and Dr. Hatejikmana, I think Elise. That's group 1. You go to autopsy in the morning, right? Then for group 2 autopsy, that will start with forensic anthropology as well. Mujenga Anselm, Ishimwe Rugamba Kamarade, Shimirwa Steven, Sibomuremyi Anatori, Bugenimana Anastasie, Niyo Twiringiye Charles, Byiringiro Clever, Iminani Pence, Bayizere Janvier, Sarihungu Patrick, Ikirezi Delis, Teta Divine Kelly, Yvette Mutesi, Niwe Mucho Deborah, Nakagame Fred.
plus plus niyo guhimbazwa Ngoga Patrick, Semana Anastase, Ntakiruta Alphonse. Yego, abo ngabo muzajya muri forensic anthropology mu gitondo, and then after lunch you go to autopsy in the Kacyiru. Is that clear? Okay. Then blood pattern analysis. Ndemerwa Glory, Evode Tuyizere, Jeannette Nyirakwezi, Rurangwa Augustine, Jean-Pierre Nkuzwenayo, Gasana Paul, Uwimana Yves, Niyongabire Jean-Pierre, Ntamukunzi Gaspard, Rubagumya Francis, Kayesu Janet, Irumva Serge, Girimbabazi Nadege, Niyibizi Emmanuel na Benda Clement. Yo ni blood pattern analysis ni Kacyiru. Saa 2 kuba muri yo. Then PTSD na GBV both combined. Umurerwa Anik, Muragijimana Isaie, Ishimwe Honore, Girinshuti Agape, Mandela Ntwali Fidel, Jacques Karekezi, Rwanashyaka Emmanuel, Mugisha Jean Paul, Murenzi Patrick, Niyongamije Ezekiel, Niyo Mugaba Fulgence, Rwenge Matthias, na Dr. Nshimimana Auguste. Abo ni PTSD, they will be here. Then cornea toxicology. Toxicology ni Kacyiru. Habimana Remy, Ngoga Sezibera, Umurungi Kolozi Sandra, Bakanibona Eden, Ahishakiye Gilbert, Mutambyi, Niyonsenga Jean-Marie Vianney, Kaneza Esperance, Twiringiyimana Pacifique, Kabira Elise Sandra, Nshutiyimana Jean-Claude, Uwimana Yves, Ngezahayo Egide, sinzi niba ari Hawiyezu Jean Pierre, Umutesi Alice, Ikuzwe Ange, niyo guhimbazwa Ngoga Patrick, toxicology. Then for cornea, there is Thomas Habanabakize, Adam Jean-Marie Vianney, Mutamba Sona, Byiringiro Evariste, Commander Ange Pascal. Abiyumva kabiri, I don't know, you have to choose one. That was a mistake. Before you go, you let us know. Niba wiyumvishije habiri, you let me know kugira ngo twaprove kuri mwe, otherwise you can't go there in the same. Ntago wakwicamo ibice bibiri ahantu hamwe. Uw'urugori Nishimwe Delphine, Ufitinema Yolande, Uwemeyimana Yvonne, Nishimwe Aline, Kananga Wilson, Tuyishime Esperance, Niyongira Norbel, Onsfield Kazitunga, Uwiringiyimana Faustin, Girimbabazi Nadege,
Rurabiyaka Joseph, Rubagumya Francis again, Izabayo Mboyi Richard, Bazumutima, Niwe Mucyo Deborah, na Ruzindana Denise. Yeah. Harutiyumvisha? Anyone who didn't hear their names? Okay. You can remain and then we put you into workshops that you want. Anyone who had a workshop that they prefer another one or switch? Then the other thing is that on Wednesday, on Thursday, those that you would have not gone into, like if you have been maybe in cornea and you want to go to autopsy, you can go there. And if you have been in blood pattern analysis and you want to go to autopsy, you have to you can let us know and then we can switch you there. Does this sound good? Good. So see you. Please make sure you can. But